we're moving on to mechanical priming. So today...
So you're not audible.
Sorry? Yep. Please. Okay. Okay. So, and as I said, then of course we moved on to the case of the chemical priming. So today the last two lectures this morning will be on the two other main fields of priming. So we will first cover principles and examples of biological priming. Actually, this section is the one that I am least, I would say, in my group anyway, active in, although we do also handle such approaches, but it is becoming increasingly important, especially along with the interest of the big companies to promote biostimulant technologies. A big part of the biostimulant story is in fact based on microbials. So this is attracting a lot of industrial attention. And then we will have a brief break after I finish the specific section in order to continue with the last one, which is dealing with the use of nanomaterials in priming technologies. In the afternoon, we will resume by doing the IP discussion, the IP workshop, and then we will also have a face-to-face interaction with questions regarding the series of lectures, but also maybe practical questions you might have with your experimental setups in your doctoral thesis. So, as I said, this is an emerging field that is attracting loads and loads of attention. Of course, as a personal comment, as I said, I think that reproducibility as well as pinpointing of the exact mode of action is indeed much more challenging in such approaches, exactly because it is usually involving different combinations of either microorganisms or microorganisms including organic acids, fulvic acids, humic acids, and so on, when we're talking about biostimulants. So it is much more challenging to carry out, rather than conventional chemical priming with a single chemical molecule. So effectively, this is the concept of biopriming in agriculture, which is indeed a great sustainable approach for crop enhancement. And as I said, lately is attracting more and more attention as an eco-friendly but also cost-effective technology that has been shown to enhance growth and improve yield, both under control as well as stress conditions. So in order to carry out the biopriming, of course, this involves the application of beneficial microorganisms. And this can be done, of course, in seeds, but also in this can also be applied exogenously in seedlings growing in the field or the glasshouse, thus stimulating plant production while maintaining environmental balance. So there we effectively take advantage of the tripartite plant-soil microbial interactions, where we have the matrix of plants, the rhizospheric growth of the plants in the soil, and then we also have, of course, the presence of both bacterial populations as well as potentially arbuscular mycorrhizal fungi. So we're always talking about symbiosis, of course, in which case the symbiosis is manifested. Symbiosis in plants are very common anyway. I mean, you're all aware of nitrogen-fixing bacteria, and in this case, the microbe will actually benefit by obtaining nutrients, whereas the plant will benefit from obtaining nitrogen and in assimilable form. But you also can get that, of course, in the case of arbuscular mycorrhizal fungi, which again assists in the mineral nutrition of the plant and the water uptake, with the fungus again gaining on obtaining carbohydrates for its own energy demands, because both of these are heterotrophic organisms. So, as you can see, there's many roles that are proposed to microorganisms, including nitrogen fixation, as mentioned before, phosphorus mobilization, but is also linked with the AMF story, potassium solubilization, iron mobilization. There has been serious indications of pest and disease suppression. We have organic matter enhancement, but also soil aggregation. So it has been shown to have multiple benefits on various studies that include all major categories of crops. So there is ample publication literature showing beneficial effects in cereals such as wheat and maize. It's also been shown extensively to be used in the case of horticultural crops, vegetable crops such as tomato. This is mainly the case for biostimulant commercial products, but also pulse crops. And the use of bacterial strains in seed biopriming in specific has been particularly effective. Now, a great part of the story is the benefits on environmental impact, as bioinoculants used in seed biopriming, of course, accumulate in the rhizosphere, and thus they promote biochemical nutrient cycling, enzymatic activity, as well as facilitating better nutrient translocation. And this ultimately contributes to improved crop performance, especially under climate change scenarios. So, as key points that have been actually observed and recorded in the literature, when it comes to the use of PGPMs specifically for stress amelioration, other components that were not mentioned before do include the production of exopolysaccharides, but also osmoprotective molecules. They possess the AAC deaminase, which is involved in the ethylene cycle, of course, and there is also elements involved in the ROS detoxification. In terms of mitigation of biotic stressors, this has been shown to be involved in the induction of systemic resistance. There is signs of antibiosis, but of course there is also competition for nutrients in certain cases. And in terms of PGPMs as enhancers of soil fertility, as I mentioned before, there is a very broad list where this is applicable. So the bioinoculants thank you. The bioinoculants, as I said, usually contain bacterial and fungal compounds, but we have somehow correlated the concept of biopriming with that of the biostimulation and biofertilization. And of course, in biostimulants in specific, you often also see in those formulations algal extracts like Ascophyllum nodosum, which is extremely common in the literature, and basically main seaweed extracts, so brown algae especially, are used for these biopriming solutions. Also, however, in biopriming, it is very oftenly used to have organic acids, such as humic and fulvic acids, or combination of those two. But also, it is very common to use biopolymers that have great benefits, such as chitosan-based solutions. So all of these act as biostimulants, as I mentioned before, and promote plant growth and enhance stress responses. The biostimulant market right now is a multi-billion-dollar market, and it is heavily promoted by national funding agencies throughout the world. So it is a field that is, as I said, attracting loads and loads of attention. So seed biopriming techniques utilizing microbes as well as other biostimulants have demonstrated great increases in seed germination rates as well as uniformity in germination, seed vigor, and generally improved viability of the crop. So the application, of course, can be done with many ways. The same logic applies as with the chemical priming, so this could be carried out by foliar spraying, by soil application through soil irrigation, root watering, but also, of course, lately a lot of work is being done on seed biopriming in specific. The problem with seed biopriming, of course, is that especially when you use, for example, polymers like chitosan, if you actually choose to use also chemicals in addition to that, then you just have to be really careful so as to see if there is biocompatibility between the various components, because often the microorganisms that are used often can be inhibited in their growth by the other compounds such as the chitosan, or effectively maybe even chemicals such as melatonin and other priming agents. So what we have seen following the application of these biopriming agents, we've seen commonly induced gene expression levels of main antioxidants, and most likely this covers superoxide dismutases, catalases, and ascorbate peroxidases. We have seen upregulation of a number of nutrient transporters, ion transporters, but also, of course, the activation of numerous stress-related genes. At the cellular level, main observations include higher photosynthetic rates, increased chlorophyll content, so pigment biosynthesis is increased. We have improvements in both primary as well as secondary metabolism, and increased overall antioxidant capacity. And phenotypically, which is again, as I say, the most important observation that you could do for a successful priming approach, is that there's observations of improved growth of both root and shoot, higher yield parameters, which is effectively the most important scenario, improved flowering, better fruit nutritional quality. There's also indications of improvements in quality traits, higher leaf number and vigor, increased somatal conductance, but also increased relative water content and higher nutrient acquisition, uptake, as well as transportation. So in the case, for example, of following seed biopriming, this is done with several scenarios. Most likely the main application method would involve soaking, although sometimes, of course, coatings are actually employed to carry out the biopriming. So following this biopriming, there is ample indications that this leads to the activation of hormonal signaling pathways, including jasmonic acid signaling, that actually leads to the immune activation and induced systemic resistance. So overall, there is the concept of the so-called vertical transmission of seed microbiomes and seed biopriming towards the maintenance of plant fitness, which is of course also beneficial for the overall ecosystem, agro-ecosystem function. So in addition to the health and the vigor of the seedlings, of course we get, as I said, the lower pest incidence, but this has indirect also benefits, because this also leads to lower input for agrochemicals, and this in its turn actually has beneficial effects on the environmental load. So the effects are both direct as well as indirect. At the seed level, we observe a balanced seed microbiome. Upon germination of the seedling, we have the establishment of the microbial community, and during tillering and upon flowering and harvest, of course, we achieve faster growth rates of the seedling. We have abundant, ample exudation of root-sourced metabolites, and at the end of the day, of course, we have the establishment of a healthy, stable root microbiome. This, of course, by itself is associated and interacts with the soil microbiome, which is quite extensive, ultimately assisting, as I said, in improved function that at the end of the day leads to higher yields. Below ground activity, of course, is equally important as above ground activity, and a big part of the story is actually the high amount of soil organic matter. So there's been studies that have shown use of these agents against many different stressors, mainly focusing, of course, on drought, salinity, but lately also on heavy metal stress. And this is usually correlated with reduced metal uptake, but also the removal of heavy metals through biosorption. This is another way of naturally detoxifying heavily contaminated soils that could actually be relevant in regions that are close to mining fields and so on. So this is also a way of leading towards a phytoremediation approach that is also quite biologically relevant. In the case of salinity in specific, when applying those microbial agents, there's been heavy implications that in the response of the hormonal regulation, we actually have heavy regulation, specifically of auxins, as well as gibberellins. And in the case of drought, as I said before, we have involvements of the ethylene pathway in specific. We have heavy productivity biosynthesis of proline, so osmolytic behavior, but also we have the involvement again of auxins, of indole acetic acid. Now, the efficiency of plant growth promoting bacteria in bioenhancement greatly relies on the precise application. And actually, last September, I delivered a speech in a biostimulants conference in Belgium that was heavily attended by companies, biostimulant companies. And a lot of them presented their own research. So they had their own products. Of course, it was academics as well as the industry, which is the ideal scenario. And many of them actually presented their own products. What became increasingly clear is that they were stressing out the importance, as you stressed out as well in questions, of the precise timing. Basically stating that the timing, and not so much necessarily the precise concentration, is crucial to make or break. Basically, they claimed, because in the field, often biostimulants and biopriming solutions have claims of not working, of being temperamental, of generally not acting as stably as single molecule approaches, like chemical approaches. And according to the companies, this is mainly because of the fact that precise timing is of huge essence. Okay? So effectively, even being a few days late in the application could effectively really have specific effects on the efficiency of the technology. And this is usually correlated with the complexity of the solutions that are usually comprising these biostimulant products. Now, common practices for the application, of course, include the soaking of seeds, as I said, in water. And this is for hydration, of course, before mixing with the bacteria, or hydrating seeds first, and then treating them with the microbial suspension. Of course, nowadays, the most exciting field is related to what is following afterwards, which is the nanomaterial story, because basically we nowadays have loads of solutions that are carrier-based microbial inoculants, and for that, this can be done, of course, with functionalized carriers with chemicals, but also many people now use plant growth promoting strains. And these inoculants have shown the potential to increase crop yield by almost 12 to 20%. Companies have mentioned that a product, a biopriming product, can become marketable if we get increases in yield higher than 6 to 7%. So the threshold for the industry for a biostimulant product that you might be developing in your lab, so as to have potential for commercialization, should have a 6 to 7% increase. And if you ask me, this is really achievable. The threshold that is acceptable in the market for biostimulants, for biopriming solutions, is actually much lower than the threshold that is usually sought for by industry for chemical priming solutions or agrochemical priming solutions, where the equivalent number that I was quoted for chemical-based or synthetic chemical-based solutions was 12 to 14% onwards. So we're talking about almost twice as much efficiency. Anything less than that, 10, 12%, they won't even bother to examine. Okay? Whereas with the biostimulants, they have much greater flexibility. And this is linked with the public consensus, with the approval rates. That people are willing to use something having less yield gain on the concept of being environmentally friendly, green, sustainable agriculture-based approach, and so on. Okay? So this is good news for people who are working with biopriming approaches, of course. The other thing, of course, that is good news in comparison with agrochemical approaches for protection is that the stringiness, stringiness is like how strictly they have to test the products. The gravity of testing is much more relaxed for the biostimulant products. The portfolio of data that has to be produced is much less pronounced. And generally, if I remember well, it was mentioned that for an agrochemical-based solution to be marketed, you need usually about a total period of about 12 years from conception and testing to validation to commercialization, whereas this number goes down to about 4 years in the case of biostimulants. And if we're talking about biofertilizers, this can go even further lower, drop even lower. So there is much more flexibility also in terms of how strict the regulatory part of the testing is. For the carrier-based microbial inoculants, however, as I mentioned before, the other thing you should remember, and I will repeat that again later on because it's important, is that when you do use carrier-based inoculants, a very important part, of course, is the biocompatibility. So you have to make sure that the carrier and the inoculant are biocompatible, because it is, as I said, often seen. I mean, in my case, we've done coating of seeds with polymers that was functionalized with natural metabolites, but actually we would see that the establishment of the bacterial populations was actually inhibited due to the activity of the priming agent. So you do need a lot of standardizations and optimizations to actually achieve ideal combinations of concentrations in order to make it work. It's not easy. Of course, we also have the production of siderophores, as I said. Antibiotics are often seen in some of the cases of the microorganisms that are being produced, but also we have observed the production of vitamins following the application of the bioinoculants. So, as I said, of course, it's important to always remember that for the seed story, you have to also be concerned about surface sterilization prior to your application, prior to your soaking. The other thing you have to ensure is that you will actually ideally avoid the plumule emergence before the seed is sown, because in certain cases with the pre-treatment of the seed, sometimes you do get breaking of dormancy, so this will actually be an unwanted side effect. So seed pre-treatment with the plant growth promoting microbes enhances plant immunity and efficiency, mainly under stressful conditions. And this approach develops specialized features in association with plants in order to combat the negative consequences of abiotic stress. So this is achieved through alterations in physiological, chemical, but also in the endogenous biological responses, including metal chelation as I said, hormonal homeostasis, and enhanced resilience through root and shoot growth simulation. So I have, as you see, several figures in order to highlight the importance of these microorganisms. The types now of biostimulants used in biopriming agents in agriculture are quite diverse, and it is important to remember that the terminology or the accepted categories of biostimulants have often been altered since their great development in the early 2010s. So far there's been 3 or 4 new definitions that were altering what we consider biostimulants. So this is actually a very fluid field, I would say. There is people that are considered to be experts in the field, and basically my personal comment is that often people come up with their own definitions. So there is kind of an insecurity in the field that for me is quite stressful as a researcher. Okay, what is thought to be a biostimulant and what is not. Okay, there is, as I said, people that are actually creating European guidelines, such as Professor Dujardin in Belgium, and these are generally considered as the ones that we follow as directives, but there is people who actually have alternate claims, and this really confuses approaches. The main categories, however, for this, as I mentioned before, is, as I said, the ones that are basically accepted by the majority of the people, is basically extracts, as I mentioned before. So this is really small, so I have to take off my glasses. So the main ones, of course, are the seaweed-derived ones, and the main seaweed-derived ones are the brown seaweed-derived ones that produce biostimulant solutions. We have, of course, polyphenols that are actually used in such cases as well. We have chitosan, which is quite commonly used because it is also shown to have antimicrobial effects. Other polymers, such as hydrogels, are not so strong on the antimicrobial story, but for example, they really improve soil water retention capacity. So they have other benefits, whereas other polymers have other ones. You choose the one that works best for your story. And of course, you get plant-derived bioactive substances. And the thing that I think is really exciting, and this is something we also work in my group, is that nowadays, heavily promoted research is also relevant to identifying indigenous plants. So basically, for you to identify local cultivars or species indigenous to India, okay, usually MAPs, medicinal aromatic plants, that are very rich in secondary metabolites, and effectively carrying out extraction of secondary metabolites and using those plant extracts as biostimulatory solutions for the improvement of growth. This is a field that is attracting loads and loads of attention, of course because of its green nature, but also because of the promotion of national germplasm that is also extremely important. You know, in our case, in our project, which is funded by our national funding agency, RIF, we have identified, for example, 12 indigenous species of plants that are found in different geographical regions in Cyprus that were known to have ethnopharmacological importance. They were commonly used by our grandmothers and our grandparents to fight off infections and so on and so on. So we literally, what we did is we carried out in the project sampling of different organs from different regions as well, because of course altitude, the geographical positioning also makes a difference on the secondary metabolite profile. And basically then we optimized extraction protocols with analytical chemists so as to actually get optimal recovery of these metabolites. Of course, we carried out extensive LCMS analysis to map all of those secondary metabolites. And then the next step, of course, was to carry out both in vitro bioassays for antimicrobial activity, but also using human cancer cells in vitro testing for biomedical applications, which is also very important. And the other side was the side that was done in my group, where we actually carried out the plant growth promoting and stress protecting function. And these approaches are becoming much more and more important to actually have plant-based solutions, mainly focusing in each region's reality. Because I am sure that in India there is some wonderful flora, some amazingly diverse species that are local to the regions and that actually have great properties. My advice is that this should be heavily promoted. This is a field of research that I think that younger scientists could easily pursue. So, of course, we have a big part of the story is the microbial inoculants, which is directly linked with the biological priming story we're talking about. And this includes, of course, all sorts of symbionts, such as beneficial bacteria, but also beneficial fungi. You have the presence of acids, such as humic substances, fulvic acids, humins, but also, of course, very commonly you have cocktails of amino acids. Most of the commercial products that are sold in the market are basically cocktails of amino acids and other organic acids. And of course, you also have other inorganic salts, such as antitranspirants, protein hydrolysates, and enzymatic extracts. Other things that can be used, of course, is protein hydrolysates. Many people lead to technologies of protein hydrolysates, and as a source, for example, it's very common to use legumes. So this is again something that could be of relevance for local standards, because I think that legumes are an important part of the local production system. Yes. And of course, the above are generally the four main pillars of biostimulant products that lead to the improved stimulation of plant growth and improved tolerance to abiotic stress. But for a biostimulant to be considered a biostimulant according to the latest directives, this also has to be linked with nutrition. Nutrition has to be part of the improved story. Otherwise, the classification of the biostimulant component is really not achievable. So the nutrient element has to be part of the overall portfolio of data that are being produced, and an improvement in that, of course, you know. So first of all, for the microbial inoculants, of course, this encompasses microorganisms that promote plant growth, and the role they have is, of course, to improve overall field performance, increasing vitality, but as I mentioned before, through the improvement of nutritional characteristics. In terms of application, this is used in soil pretreatment, but also in seed, quite commonly. Then, of course, you have the biopolymers, which are natural polymers that are derived from living organisms. For example, chitosan is very commonly produced from shrimp. You actually get the processing of shrimp, and with that you get your lower high molecular weight chitosan. So again, in this case, the role is through the regulation of physiological processes, again towards accelerated growth and stress tolerance. And again, the application can be both at the seed level but also at the soil level. And for the nanomaterial story that will follow later, nowadays it's very common to obtain chitosan nanoparticles, so actually go for nano-size particles of chitosan through specific procedures in the lab that are basically conjugated with specific chemicals. And it is also very common to use encapsulants. So chitosan is very commonly in the literature used along with TPP, and this creates effectively something you could imagine as a microbubble, okay, in which your priming agent of interest can be entrapped. And with that, you can, of course, continue and also carry out application in a seedling through exogenous application of the material. So in terms of the bio-based polymers, you can see that again the actual source and the diversity is huge of what could be effectively used. Of course, in addition to the one I mentioned before, there is sodium alginate based solutions for coatings. There is many different types that are being examined right now. It's like cellulose based, there is many different things. So you can actually have them synthesized from bio-derived monomers such as PLA, polylactic acid, and other polyesters. You can have them directly extracted from biomass, okay, which is effectively the most common approach for somebody to work with the biopolymers. This can actually be through protein extract source, polysaccharidic source, or lipidic source. In the case of lipids, this could be represented through, for example, beeswax or free fatty acids. From the polysaccharides, this is of course related as a source from starch or starch derivatives. Cellulose, and this is actually attracting quite a lot of attention nowadays in research. Yeah.
Generally, we are using polymer as a carrier material too. So, what I'm asking, if I'm using any commercial polymer as a carrier material, is there same biostimulant property as the extra?
It remains to be tested. Generally, this is a general advice. How should I put this? There is never the easy way. I realize that for young scientists it's always preferable to think or be told of an approach that will be guaranteed, fail-proof, this is better, this is not better. It doesn't work like that. Questions like this are actually, and don't get me wrong, invalid questions. This is not something that you can actually determine like that. Any sort of material, I really strongly believe, could be ineffective, but at the same time, upon proper experimentation, become effective. I don't know if you understand what I'm saying. So it's not like you can say that commercially obtained material doesn't work. Biologically sourced material does, or vice versa. Everything is pending examination. I mean, again, I know this is not the answer you want. It's not the answer you were hoping for, but this is the reality. Whatever it is, it remains down to be tested. You go down in your lab, you actually put down the materials, you test them, and you actually check for the efficiency. Actually, the question, is it commercially sourced or is it biologically sourced? What you should be asking me is about what should we go for? Both of them represent solutions. It all depends on speed, on cost, on potentially improved activity. You could see through testing that biologically sourced material that is alginate based, for example, works better in your setup, in your specific interaction of crop and stress, in whatever story you're trying to say. So this is what you would go for. On the other hand, you could say that buying low molecular weight chitosan from Sigma Aldrich is good, but it is super expensive. And I know, what I'm trying to say is that I know people, I have collaborators, for example, in my lab I have also researchers from Iran, and they told me that in Iran they actually use chitosan in their approaches quite often, but because of the cost of the material, they've actually optimized protocols where they make their own chitosan from shrimp waste, from shrimp. And because for them it is much more cost effective to actually go through this procedure. So that's why I'm saying the answer is relevant. Everything is down to each person's reality. So there could be improved performance, but sometimes the improved performance could not be the realistic approach for you to follow for other reasons. I hope you understand what I'm trying to say. So in terms of the molecules, if the polymer, like practically speaking, I've actually asked to obtain some of that shrimp, naturally produced shrimp-sourced chitosan. So hopefully I will get some soon. I'm also curious to actually test it side by side with some commercially sourced chitosan we get from Sigma, for example, and to also test the homemade recipe that the professor is doing in Iran. I'm also curious. But in my mind, my expectation is that I don't expect to see huge differences. But I still want to do it. I still want to test it. It remains to be seen. This is my response to you. Even in my case, I never kind of rely on labels or whatever. It is all down for you to test and to see what will work better. Unfortunately, a lot of the materials are not cheap. This is the reality. And this is not just for polymers. This is for everything. And in most cases, for most labs, this is not easy to sustain. Costing-wise, because in priming technologies, cost is the #1 thing that the company will ask you for. Actually, again, it is not so much, Vasily, how well does it work? Because if you reach your 6, 7, 8% increase, it ticks the box to be of potential interest. And as I said, reaching that improvement is not the most difficult thing on the planet. It's not easy, but it is achievable. But afterwards, the company will have a million other questions to ask. One being the costs of your approach, for example, because they also have to check cost-benefit analysis for something to be marketable. NOS aspirin, I'm going back to the molecule that we use that I'm trying to commercialize. As a joke now, as a game, who wants to give me a guess of how much it costs to formulate 2 grams of active ingredient? 2 grams of active ingredient. The cost we pay to a company to formulate this compound, this hybrid donor I talked about. Just for you to understand the stress we're going through. But of course, afterwards I will explain that although the number, of course, will be high, you can understand that I'm going for a high number, there's ways to actually fix that. The cost of 2 grams of formulation is $3,000 for 2 grams. So actually one of the reasons, again, I'm just being blatantly honest with you, one of the reasons why we work heavily with seed coatings now and seed priming is also because of that. On the way, we saw that it's also very efficient, but initially, one of the reasons why we shifted from the plant application, the foliar sprays, for example, to that, was because when companies were coming and we were saying, like, let's try and spray and do and whatever, the cost of application for a field of wheat was tremendously higher compared to what it would take to prime the seeds in the lab, because usually we would need about 50 ml of volume of dissolved NOS to treat 1,000 seeds. So if you do the math, the cost per seed per plant would be very low. We're talking about point something cents per plant. For plants, you would need huge volumes of spraying. So the cost would actually make a huge difference for the approach. So costing, yes, costing is a big issue as well that you always have to remember. The other thing. Even though something that you develop, if it is expensive, of course you have to remember that upscaling will drop the price. If you propose like a solution to an industry that is relatively expensive, although biopriming is usually not expensive in comparison to chemical priming, a big company will be able to upscale, and upscaling will always bring the price down. Like, for example, in our case, the company that is formulating the 2 grams of NOS aspirin, we get the quote of 3,000. So as the number drops to what? It's per gram, that's 1,500. Upscaling this to 50 kilos, it would bring the cost down per gram from $1,500 to $14. So basically, what is it, like 100th? What is it? 1/100th of the price. So upscaling is always a good way to, of course, to reduce the cost. So don't panic. If something in the lab, in lab scale experiments, is a bit intimidating in the cost-benefit analysis, in the long run, on the industrial scale, there are ways to actually improve the situation, of course. So, moving on. As I said, we have cellulose and cellulose-derived sourced material. We have gums, alginates, such as which I work as well with. We also use sodium alginate-based polymers in my group. And actually, one of our patents is based on sodium alginate solutions. Of course, we have the use of chitin and especially chitosan. And of course, a major source for the biostimulants and the ones that are sold in the market are based on proteins. So you have, of course, animal proteins such as casein, gelatin, and collagen, but also plant-based proteins such as soya-based proteins, pumpkin proteins, wheat gluten, and zein as well. The movement lately I see in the market is mainly trying to get protein sourced from leguminous crops. So before I mentioned also in the biopriming solutions the algal extracts. So these are extracts from algae, as the word suggests. They are also shown to improve plant growth, enhance stress tolerance, and contribute to overall vitality. And they are also suitable for seed as well as soil pretreatment, but also foliar, of course. Foliar should not be excluded. And you have nitrogen derivatives that are compounds that contain nitrogen, and these enhance nitrogen availability, contributing to improved plant growth. And again, this can be applied in virtually most forms. So the key mechanisms that are targeted specifically by algal-based biostimulants, and actually many of the commercial products usually do contain Ascophyllum nodosum extracts. It's extremely common to find this in the market. So in terms of whole plant responses, the main responses that are usually recorded involve, of course, improved ROS scavenging, improved membrane stability and osmoprotection. In terms of shoot targets, we have stomatal regulation as well as xylem, improved xylem hydraulic conductance. In the case of the root, when we're talking about organ-specific responses, we have alterations in root zone water availability and also root ethylene and auxin levels. Hormones can be commonly used. This is why, for example, melatonin is a good solution, because this could fall under the biopriming approach. Organic compounds, therefore, that act as signal molecules, and we talked about this extensively in the previous days. These again regulate growth development, but also increase stress tolerance. Application can be done again on any organ. Many commercial products contain humic acids that are organic acids that are actually sourced from decomposed organic matter. And this improves mainly nutrient availability, but also the soil structure. And this is, in addition to using it in coatings of seeds, this is often used as a soil amendment, the humic forms. So overall, here you can see a diagram from a review on the use of humic acids. So you can see here that you have different forms of humic acid. You have low molecular weight and high molecular weight humic acid, where the function is actually quite differentiated, where in the case of the low molecular weight ones, we do observe the co-transport of nutrients via the plasmalemma, whereas in the other case, the high molecular weight, we do have regulation of proton pumps largely, which of course secondarily also affects the nutrient transport. And the humic acids, of course, as a chemical structure, they contain aliphatic groups, aromatic groups, phenolic groups, or carboxylic groups. So in the case of the latter, we have a direct improvement of soil physical and chemical properties. In the case of the former, we have stimulation of root growth, improved nitrogen uptake, and also soluble sugar production. But at the end of the day, the end result is the same, the increased agronomic performance, but also improved quality parameters. So you can also get MAP extracts, herb extracts, as I mentioned before, which is again applicable in any organ. But also you can use other organic chemicals, so various organic compounds that have biostimulant properties. And these are mainly applied in the soil actually. So microbial seed priming is a long-term solution for water scarcity. As it generates phytohormones, it is involved in the biosynthesis of ACC deaminase, exopolysaccharides, antioxidants, but also we have observed the activation of stress-responsive genes specifically for drought tolerance. And in the case of the exopolysaccharides, they have been shown to benefit soil health because they aggregate soil molecules, they preserve the water balance, and they ultimately enhance plant survival under adverse conditions. So this is a brief representation of the microbial sourced EPS. So you have different forms. Again, these are quite diverse categories. Of course, I'm giving you the brief outline in each case, but each subsection has vast literature behind it. So in the case of the EPS, we have homo as well as hetero exopolysaccharides that are differentiated between single type as well as different type monosaccharides. So the list, as you can see, and we're not going to go through it in huge detail, is quite broad, quite diverse, spanning from glucans to fructans to polygalactans, and then of course also covering other derivatives as well as glucose, galactose, and rhamnose. So a very broad range of exopolysaccharides. Specific examples are actually also listed at the bottom here, but each and every one of these exopolysaccharides is actually mapped, and we do know its exact chemical structure but also biological activity. Now, this is a great field. The microbial exopolysaccharides are very commonly used in the industry in many diverse functions. So you see here that the list is quite huge. It is used commonly in the food industry as well as in cosmetics to control viscosity and modify flow. It is improving, of course, texture as well, which is very important for the perception of ultimately also affecting taste in a way. It's thickeners, softeners, suspending agents, but also they improve the freeze-thaw stability. They're used in the pharmaceutical industry for the encapsulation of drug delivery systems mainly, but also they're using as coatings for lotions. They're used in the mobilization of enzymes, blood volume expanders, as well as for the chemical modification of pharmaceutical drugs. Now, in terms of general industrial activity, they're used as lubricants, they're used as dye, as well as pigment, overall pigment suspensors, and they're also used as rheology modifiers for the fluidity of the materials. In dairy technology, they're used as stabilizers, texturizers, and also to improve elasticity. You can see here that there is also functions in medicine where it is used directly for the maintenance of gut health, as anticoagulants for the blood, as laxatives and so on. And of course they have very general antimicrobial and anti-inflammatory and anti-cancer activity, so they pretty much are used everywhere. Now, specifically for the stress story, a common, of course, stressor would be that of salinity. So they have been very extensively shown to alleviate salt stress damage. So this is mainly done by, of course, improvements, as I said before, in nutrient uptake that results ultimately in ion homeostasis. There is, of course, also the activation of the antioxidant apparatus, so we have a reduction in oxidative stress levels. And the other major modus operandi is that of the production of osmolytes, so we have enhancement of osmoprotection in the cell. So this is done both through the direct activity of the microorganisms, but also through the induced response of the plant that actually does exudate through the root several metabolites that are actually involved in this protective response. In terms of drought, this is also extremely commonly employed. We have seen in multiple papers beneficial microbes increasing drought tolerance and water use efficiency. This can be done with both epiphytic as well as endophytic bacteria on the aerial tissues of the plant. So we have effectively the involvement of the phylospheric microbiome. But of course, most commonly, most likely, we also have the application of rhizospheric microbia that are actually functional in the root and modify host responses. And this can include, as I said, also bacterial populations, both natural, but it's quite common nowadays to also identify microorganisms mainly from composts or some cultures where plants are naturally tolerant to conditions. For example, in halophytic plants near the sea, there is very often extraction protocols where you can actually identify microorganisms that are involved in the response. And following the mapping of each one of those bacteria, initially in vitro and then in planta, it's also very common to then try to formulate your own ideal cocktails of microorganisms, creating the so-called syncoms. The syncoms are the synthetic communities. So you can identify individual strains and you can then put them together in an attempt to create a super cocktail of microorganisms that will actually lead to further improved performance. So yes, this includes also AMF, of course, but also in some cases, although not very common, viruses. And the main protection is achieved through the top 4 categories of alteration in signal networks. So alterations in the reactive species content, regulation of phytohormones, obviously, enhancement of the antioxidant apparatus, but mainly osmotic adjustment. Secondarily, there is other functions that are linked with this protection, such as VOC production, increased WUE as well as NUE, triggering of induced systemic resistance, and so on and so on. Now, also several bacteria, mainly Pseudomonas strains as well as Bacillus strains, have been shown to also alter heavy metal mobilization. This is also very common. And extracellular polymeric substances basically help a lot in limiting heavy metal translocation, thus offering a natural solution for soil health. Now, in terms of seed coatings in particular for biostimulants, cycrotolerant microbes can be used in seed priming to improve plant responses to low temperature. Cycro is the Greek word for cold. So this is the etymology for this category. And the integration of the microbial inoculation and signaling chemicals in seed priming is shown to enhance stress tolerance. Now, seeds bioprimed with PGPRs experience a big variety of cellular changes, although the cellular constructions of the organelles remain largely unchanged. Okay, so anatomically speaking, we don't see major alterations. It is more at the cellular signaling level. Some PGPRs have also been shown to influence the RSA, the root system architecture, the root system morphology, including elongation and also changes in the composition of the cell wall. Okay, and specifically, the specific microbiome, which is Azospirillum lipoferum, strain CRT1, has been shown to lower lignin content, to promote cell elongation, and to increase root length. Of course, the lowering of the lignin content, this is just like a thought that came now spontaneously. Such microorganisms could potentially also be involved, since it affects lignification, in biofuel studies, because of course the lignin content is a parameter that actually affects how easily you can actually extract the polysaccharides to actually obtain your biofuel of interest. So this sounds like something that could effectively also be employed for biotechnological approaches of improved biofuel productivity, let's say. And as I said, this strain promotes cell elongation but increases actually root length, especially the taproot of dicotyledonous crops. And we have also seen in Brassica species increased nutrient uptake following specifically Achromobacter species inoculation. So here you see phenotypes in cases where we have rhizobial symbiosis in legumes, and here, for example, you see that you have very nice pronounced growth of the root with rhizobial presence in comparison with the uninoculated, unnodulated root. You actually can have improvements in root growth following the use inoculation of other PGPRs, but you can see that often through synergism, the addition of PGPRs to your rhizobial populations, you can have very extensive alterations and further growth in the root system, which is quite dramatic, as you can see. So the employment, summarizing basically the employment of these PGPRs, we have, as I mentioned before, a number of benefits to both a plant level, but also as well as the whole ecosystem level. But mainly, a lot of the effects are initially observed in terms of root traits in specific. So often we see changes in root branching. So the root system architecture, as I mentioned before, the number and length of the lateral roots also has a big effect. The length and the number of the root hairs, which is also very important. There could be the formation of biofilms on the root surfaces. But also, of course, there is main effects on the taproot, on the main root length and the girth. And there is also alterations in the root exudation patterns, on the profile of the secondary metabolites that are actually being exudated in the rhizosphere. And this is just a diagrammatic representation, just a depiction of how and where these processes are taking place. Now, we mentioned before also the concept of ISR. Modifications in the cell wall also do induce systemic resistance through, of course, signal pathways. And there's been cases using specifically Bacillus subtilis strain GB03. This is quite commonly used in the literature, where this led to a global transcriptional regulation of the plants that were inoculated that actually resulted in VOC production. So we have major production of volatile signals, but also changes in cell wall composition and ultimately cell wall expansion. And these VOCs that are actually being released play a role towards both induced systemic resistance, that of course is directed to pathogen protection, such as through the regulation of pathogenesis related genes, but actually it's also implicated to induce systemic tolerance. Sometimes you will also see the term of IST in addition to ISR, the main difference actually being that ISR refers to induced protection against biotic stressors, whereas IST refers to protection against abiotic stressors. Okay? And this is mainly carried out through improvements in iron uptake molecules, transporters, but mainly also in maintaining sodium homeostasis, such as through the regulation of HKT1, NHS1, and so on. So, great importance in the regulation of volatile organic compounds in the story. PGPRs, as I mentioned before, a main mode of operation is through the induction of phytohormones, mainly the auxins, cytokinins, ethylene, GA, and ABA. There is secretion by PGPRs of auxins that actually modifies the root architecture. And of course, there is involvement in cytokinin and ethylene production. Now, some plant growth promoting bacteria like Azospirillum and Arthrobacter, they produce cytokinin influencing again root architecture, and ethylene production by specifically Azospirillum also has been shown to affect plant senescence as well, also through the regulation of pigments in aerial parts. And in terms of GA production by PGPRs, various PGPRs have been shown to produce gibberellins. Actually, I don't know if you know this, but gibberellins were first discovered isolated from fungi. It was so you know the fungal disease of Bacana. It's you call it Bacana. Bacana, is it? Bacana, yeah, that's it. Yeah. So yes, so everything started from the fungal story, and then of course we moved on to actually isolate the plant-derived ones. And gibberellins, of course, have been shown to influence primary root elongation as well as lateral root extension. So they have a major role in plant growth and development. And there is, of course, the famous crosstalk where we actually have this molecular signaling that actually affects the regulation of antioxidants, affects the ROS contents, obviously. Cytokinin level regulation also is linked with downstream regulation of ABA levels. The regulation of ACC deaminase that was mentioned before actually does lead to the regulation of ethylene levels. The transcriptional regulation of HKT1 leads to serious sodium translocation, thus leading to ionic homeostasis. So everything, as you can see, is done either through transcriptional regulation of transporters or through the regulation of specific hormonal molecules, which is quite standardized. Now, understanding mechanisms linking seed priming and physiological markers is, of course, crucial for resilient crop production, and this is why PGPBs play a key role in stress mitigation. So, a very important element to consider in this protective function is the nutrient solubilization, but also the improvements in the nutrient uptake. And as I said, terminologically speaking as well, improvements in nutrient status is a prerequisite so as to actually classify a product as a biostimulatory activity one. So PGPBs have been shown largely to solubilize nutrients from organic matter, also from allelopathic substances, and this makes them afterwards more readily available to the plants. And they facilitate mineral uptake, they reduce infection, of course, and they stimulate the plant's defense system, also by regulating pathogenesis-related proteins. Now, microorganisms, in addition to the above, also produce siderophores in cases where iron is not really abundant in the soil. And siderophore-producing bacteria have been shown to alleviate heavy metal toxicity, thus leading to ultimately enhanced growth. So, in the case of heavy metal contaminated soils, as you can see, we actually have the presence of your microorganisms that actually produce the siderophores that I mentioned before. So these are bound to metal complexes, to initially insoluble metals that actually aid in their solubility, that is then much more easily taken up through membrane transporters. This can actually happen both through ligand exchange models, by direct uptake of course, but also through degradation that is actually passing through the membranes. Now, in terms of the mechanism of induced systemic tolerance, so effectively the response that is induced following abiotic stress conditions, again this is linking the IST mechanism with PGPR-derived elicitors, and these elicitors are mainly in the form again of hormones, enzymes, antioxidants, and VOCs, and microbes are known to promote plant growth through exactly this phytohormone production, heavily linking it with the ABA pathway, but also amino acid restoration. amino acid accumulation is essential in the stress response, and microbes have been shown to help stressed seedlings restore amino acid content during their recovery. And amino acid accumulation regulates osmotic adjustment and osmotic metabolism, thus contributing ultimately to stress resilience. And of course, in this story, proline plays a key part as a major osmoprotectant. So there is a recorded rise in proline content following inoculation with PGPRs that acts as an adaptive mechanism for osmotic adjustment, of course. And this increased proline content is linked to improved plant performance under harsh conditions. However, having said the above, I will mention something else. Again, this is just an advisory parenthesis. Again, don't follow everything for granted, meaning that now you have me saying that PGPRs induce proline, and this is linked with efforts to do better in the under stress conditions. It depends on how you look at things, on how you interpret the story. What do I mean? We have seen in certain cases increases in proline following priming with chemicals, but of course this was, on our case, discussed as efforts to improve the osmotic adjustment in the cell so as to withstand the osmotic pressure caused by salt in the soil or whatever, you know. We also have cases of priming approaches where the protected plants we observed had the exact opposite trend. They actually had much lower proline levels. What I'm saying is that most likely in your careers it will happen that you will observe completely contrasting observations. So it all depends on trying to make a nice story out of it. So understanding what else is happening in a holistic approach so as to create a nice mechanistic story on how things are working. In the case, for example, of the approaches where successfully primed plants had lower proline levels, like with strobilurins, we actually would see that proline would go down. So the theory is that in the stressed plants, proline goes up exactly in an ultimate attempt to regulate osmotic adjustment. Whereas in plants that are protected likely through other mechanisms, such as through maintenance of photosynthetic activity or activation of the jasmonic acid pathway linked defense and so on, this means, in my mind, that the plant has no need to spend plenty of energy to produce plenty and plenty of proline to survive. So it prefers to keep this energy and redirect it to defense strategies that are actually indeed useful for the specific condition. So what you see here, that successful approaches leads to increased proline, this is not a given. Sometimes this could be the case, sometimes based on different modes of operation of agents, this could be the exact opposite thing. And I was glad to see that this thing that we observed that made me feel a bit paranoid, because in the literature you would see people say that, whereas we would see the opposite. Recently I saw a review article published in a journal called Antioxidants, which is a relatively high impact journal, and actually the researchers there from Portugal, they made the exact same claim, that it is seen that in certain cases actually proline content under stress in priming situations will actually go down. So keep your minds open. When you get taught something by a lecturer or something, keep it in mind, consider it, but it doesn't mean that other scenarios cannot actually unveil in the future. So osmoprotectants is of course also linked with heat stress, and these also include, in addition to proline, phenolics and sugars that could be, like exopolysaccharides and sugars, that could be secreted by rhizospheric microorganisms. And recent focus on identifying the signaling pathways has been given in association with seed priming approaches, biopriming approaches in particular. So the heaviest link for this study is focusing, of course, on the phytohormonal modulation. And in addition to the above mentioned parameters, we also have enhances in protein content, in phenols, as I mentioned before, but also in flavonoids. I mentioned before that we have seen recorded increases in the secondary profile. A big part of that is in the flavonoid status, and this aids, of course, largely the plants in avoiding combating stress. In terms of ethylene in specific, high ethylene concentration leads, of course, to induction of plant senescence and stress symptoms. So when we bioprime seeds with the PGPRs, this releases the ACC deaminase as mentioned before, and this, of course, regulates ethylene production towards the mitigation of stress. The accumulation of ABA is heavily linked with cytokinin signaling, of course, and pre-sowing inoculation increases ABA accumulation that is antagonistic to the cytokinin signaling for the induced stress tolerance. And ultimately, we have an improvement in total and reduced sugar content that actually results following the biopriming of the seed. So, osmoprotection is also linked with mitochondrial activity. So, biopriming was also shown to increase other osmoprotectants like GB under water stress and salt stress, and there was effective growth that was linked to early inhibition processes in the mitochondrial activity, thus boosting energy metabolism. The secondary metabolite profile, as I said, is essential. So we have compatible solids and volatile organics from halotolerant bacteria that reduce salt stress effects. And syncoms, as I mentioned before, can enhance pigment content, so which is also linked with improvements in senescence profiles and also photosynthetic rates while reducing the oxidative damage. And you want to have like a 5-minute break? But 5 minutes though, because we have to finish by lunchtime. 5 minutes. Okay. crucial for the enhancement of stress tolerance in plants. And such biopriming basically activates known resistant stress markers through mainly ethylene, SA or JA signal pathways. And in the case of the jasmonic acid signal network, jasmonic acids and ethylene specific directly activate TFs that encode for stress and responsive genes. And specifically the jasmonic acid pathway involves activation of JAR1 that leads to the destruction of JAZ proteins. So subsequently this leads to the release, the activation of many transcription factors. So in addition to the jasmonic acid network, of course we have the involvement also of MAP kinases and the CDPKs, as well as other cyclases. So we have the PTM, the phosphorylation event of several MAPKs, as well as CDPKs that encode for various stress-responsive proteins. And under stress conditions, there is a cytosolic calcium ion content rise, as well as redox changes that basically activate the MAP kinases, thus leading to ultimate biological priming when we use Trichoderma species. Usually Trichoderma harzianum is the most common strain species that is used for biopriming. And this actually induces an accumulation of latent cellular proteins that leads to more strong immune responses. So inoculation with the PGPRs basically accelerates stress resistance at the transcriptional level, so the regulation of gene expression levels. And examples include the enhancement of drought stress responses, drought stress tolerance, in Arabidopsis thaliana when actually inoculating with Bacillus polymixa strain B2. So here you can see that upon inoculation with the bacterial strains, we have the sensing through RLKs, through receptor-like kinases. This leads to the activation of the signaling apparatus, mainly linking reactive oxygen and nitrogen species, as well as generation of calcium ions. But of course we have regulation through abscisic acid and auxin signaling in particular. This activates CDPKs as well as MAP kinases that in turn leads to the upregulation of several stress-responsive genes such as RD29B, which is effectively a LIA protein as we mentioned in the past, ABI1 and 2, which is again ABA-dependent stress response elements, and so on, that ultimately lead to the protection of the plant against drought stress. So there is also the induction of misfolded protein accumulation that is regulated through the so-called BIP genes, and it has been shown that the PGPR inoculated plants show reduced BIP gene expression levels that leads to increased stress amelioration through decreased soluble protein content and subsequent, of course, protein catabolism, proteolytic events. So there is also a regulation of program cell death events, of apoptotic events, and this is regulated by BI1 gene. This is suppressed effectively following PGPR inoculation, thus balancing BAX activity and regulating the program cell death. And experimentation in pepper plants that were actually inoculated with PGPRs revealed a suppression in BI1 gene expression levels that resulted in increased tolerance against different stressors, including salt, drought, and heavy metal stress. So there is, of course, also epigenetic modifications that take place following biopriming. Yesterday, at the end of my chemical priming, I mentioned an example, if you remember, where major DNA methylation patterns were observed in plants that were primed with Trichoderma harzianum, strain T22. So epigenetics involves heritable changes in gene expression levels without altering, of course, the genomic sequence, which is crucial for understanding priming effects, and the so-called epigenetic stress memory phenomena. So priming strategies operate on various levels, and these changes could be both short but also long-lived. And this is what renders the concept of memory. So biostimulants like trichoderma, as I mentioned before, play a major role in plant epigenetic control, and this is done mainly through histone modifications, through DNA methylation, or RNA-directed DNA methylation. And T. harzianum treatments of seeds exhibit enhanced growth post-emergence, and this is attributed to modulated gene expression levels. And here you actually see a model actually highlighting all of the previously mentioned protocols, procedures in the scheme. So, in terms of regulation at the transcriptional level, Trichoderma harzianum treated plants showed an induction of genes that were related to carbohydrate binding, glycoside hydrolases, as well as polysaccharide lyases that ultimately resulted in enhanced growth. An expression of various genes in rice plants in specific, such as those related to drought tolerance, is also shown to be induced, to upregulated, upon colonization of roots in plants by trichoderma. So there is a number of epigenetic modifiers that were identified in tomato, and specifically tomato roots, namely HAGs, AGO, and RDM1, and this was following inoculation with trichoderma that basically are known to participate in DNA methylation as well as chromatin modifications of their structures. And trichoderma actively engages in alternative splicing events that affects a number of defense gene loci. So I'm effectively closing off with the general diagram, which shows in summary and in brief the procedures that are followed upon microbial inoculation. So we have the colonization of the root, of course, when treatment is applied at the seed or soil or root level. This leads in a localized manner to enhanced growth and development of the roots, and of course this is heavily correlated with the improved uptake of water, but also the nutrients, because nutrition is achieved because of the fact that water is an excellent solvent. In the aerial parts, on the other hand, we have an increase in the photosynthetic capability of the plant and the induced improvement in growth, yield, but also nutrition and quality of the fruits, of the end product. And of course, at the cellular level, one should not forget that we talked about major modification of ROS metabolic pathway, mitigation of both biotic and abiotic stressors, as well as the systemic modulation of gene expression levels. And this was the end of the biological priming story, so that we then move on to the final lecture of the series, before in the afternoon we resume with a discussion on IP matters. And this will be dealing with the nanomaterial story. And of course, the nanomaterial story, as I mentioned before, it is of great interest, as I understand, to the local audience. It's also here in MNNIT, there's plenty of groups that actually work with nanomaterials, and from what I understood, also other attendees are interested in this, as well as in biopolymers. And as I mentioned before, of course, the nanomaterials can have a dual function. You can have direct application of the nanoparticles, either organic or inorganic, and they have been shown to have direct scavenging and protecting abilities. But in my book, the main promise of this technology lies in their potential use as smart nanocarriers, and this is thanks to their physicochemical properties, but also their nanosize that actually allows for effective delivery in subcompartments of the cell. As you know, the nanomaterials are usually within the low nanometer range. We're talking about 100, 200 nanometer range, at the point where a usual stomatal opening would be in the range of 20 to 60 micrometers. So very, very tiny size that actually does help in the easy uptake and translocation within the vascular tissues. For example, nanomaterials would be wonderful tools, in my mind, to otherwise fight off stressors that are difficult to handle. An example for that is, for example, xylem-borne diseases of plants, like verticillium. such pathogens are notoriously difficult to control with conventional agrochemicals, but actually the use of smart nanocarriers would be a wonderful opportunity through, for example, soil irrigation approaches, where by taking up these materials it would be translocated through the xylem, and then it would be directly possible to actually try to fight off those stressors. So there are specific cases where it is clear that there would be a potential advantage in such an approach. So, of course, there is major advancements in nanotechnology. Me as an editor in journals, I see there is a boom in submissions, in relevant submissions, which is very indicative of the fact that it is a very trendy field. And of course, this nanotechnology is highly relevant because it can be employed in the form of improved soil management practices. I mentioned before in advanced nanomaterials the example of hydrogels. Hydrogels are famous for being used for improvements in soil water retention capacity, for example. Of course, they can be used in nanosensing technologies as nanofertilizers, nanopesticides, nanoherbicides, and nanofungicides, but effectively also in overall water management practices. This is just to show that there is also ample literature. Recently, I was also involved in the editing of a book that is exactly in the specific subject, in engineered nanoparticles and their use in agriculture. You're welcome to check it out. It's a contribution from 11 different laboratories. So this is just one of the many, many books that are available in the literature, and I'm not even talking about research articles, which is just hundreds, if not thousands. So in terms of nanoparticles, specifically in crop protection, nanoparticles show great promise in protecting plants against stressors, and they have been shown to directly scavenge reactive oxygen species, but also to enhance photosynthetic capacity, thus contributing to ultimate stress mitigation because of improved carbohydrate reserves. As I said before, a great element of the nanomaterial story is thanks to their nano size, which ranges in the low nanometer range. So this provides a wonderful tool for easy uptake, but also they are characterized by diverse physicochemical properties that allow for effective plant interactions. And these properties render them wonderful tools to deliver chemical priming agents. So effectively, the functionalized nanomaterial story could be considered the second generation of successful chemical priming approaches. Of course, it could also involve biopriming. The only problem being what I mentioned earlier, that the only problem in that sense would be the biocompatibility concerns, that you would have to make sure that your materials are not inhibiting the microbes, because, for example, if talking about nanoparticles, it is very common to employ, for example, silver nanoparticles, which is a wonderful tool to kill off microbes. So your choice of nanomaterial has to be wise, so that it doesn't interact with biocompatibility issues. So that way we can ensure targeted delivery at an organelle level, compartmental level, and also optimal priming activity. And we do see practically that the performance of the functionalized nanomaterials is indeed improved compared to either the carrier itself, which does have priming properties, or the chemical agent we use to functionalize the material. Because, of course, in a successful experimental setup, you would need to have each and every one of all of those controls. Very extensive experimental setups, so a lot of work for the young scientists, but it is the only way to do it. Otherwise, it would be inconclusive. And there is, of course, a reduction in potential environmental impact because you would need lower amounts of active ingredient because of the smart delivery. So this effectively would have a lower environmental load, if anything. However, of course, there is always concerns about the potential effect of these nanomaterials, especially if talking about nanoparticles and especially metal nanoparticles. So in any case, of course, there is needs for research on environmental and safety concerns. So smart phytonanotechnology in agriculture basically involves the use of nanomaterials for the targeted nutrient transport, but also controlled release of agrochemicals. And the potential applications include the agrochemical delivery, the nutrient delivery for smart nanofertilization, the plant genetic breeding, because you can actually improve gene delivery through gene editing approaches with nanomaterials. This is a very booming field as well. And also, of course, the use of bio-nanosensors. And you achieve actually enhanced nutrient uptake and increased tolerance to stress, thus boosting agricultural productivity. So you can see here that the list of potential applications is pretty large, and this can be involved in both fertilization towards soil enhancement and mineral uptake, in terms of crop breeding, in combination with gene editing technologies, as I said before, of course in the crop protection industry, for improved growth and development. The stress story, abiotic stress story, is obviously always a major factor, but also it could be involved in improvements in quality traits as well as product preservation, such as through improved post-harvest performance. And here we see again a general diagram that was observed from a relatively recent review article that was actually published in Trends in Plant Science, showing a potential representation with the focus of what the authors considered to be the main points of interest, that in this case, of course, was, as I said, the target-specific delivery of products. This could, of course, be also relevant to the biomedical industry for human drugs, in any case. But actually, in this case, this was also assigned to the potential they have for in vivo labeling and imaging for real-life tracking systems using advanced microscopy as well for the nanomaterials. And this is indeed a possibility. And recently, I'm really happy to say that my group, we also were invited to submit an invited review article in Trends in Plant Science, and it was recently accepted for publication, and the topic is exactly the potential of using functionalized nanocarriers as the next generation priming technology in agriculture. Hopefully within the next month or so, or 2 months, you will be able to have a look and let me know what you think. But we're very excited about this paper. So in terms of phytonanotechnology, specifically in crop breeding approaches, we do achieve efficient gene delivery mechanisms using phytonanotechnology to demonstrate the applicability it has in plant genetic engineering. There is several groups that actually work towards improved delivery in gene editing approaches using smart nanomaterials, and therefore nanomaterials with varied properties can contribute to the improved accuracy of this delivery and therefore improved effectiveness in the approach for gene editing. Here you can see different nanoparticle classes, because actually the possibilities are vast. Experienced materials chemists and material scientists can actually create vast libraries of materials that can be tailor-made based on your needs. So enriched with metals in case you're looking for antimicrobial properties, enriched with elements such as silica or selenium, for example, if you're looking for enhanced nutrition, and so on and so on. So you can actually produce tailor-made, custom-made materials if you have proper expertise. So in terms of the nanoparticles, there is many classes that are employed. So you can have bio-inspired nanoparticles, such as ones from calcium phosphate, chitosan-based or liposomes, and the cargo they can deliver, of course, can cover both nucleic acids, either DNA or RNA, but also they can deliver specific proteins and so on. So this can be used in both animal and plant systems, obviously. You can have metallic or magnetic nanoparticles, which are usually limited mainly to gold, silver, and iron oxide nanoparticles, although the two former ones especially are quite expensive ones to work with. So many people actually resort to green synthesis because of reduced costs. My group is one of them. You of course have carbon-based nanoparticles, such as single-walled and multi-walled carbon nanotubes, the MWCNTs as we call them, and fullerenes as well. You can have silicon-based nanoparticles like mesoporous silica NPs, and I think here in India there is also quite a lot of work using mesoporous silica nanoparticles and silica spheres. You can have polymeric NPs, such as polyethylene glycol-based ones, PLGA ones, so polylactic or co-glycolic acid nanoparticles, and you can also have polyethylene EPI nanoparticles. And in terms of the cargo delivery that is mediated through the use of these nanoparticles, this can be done, as you see, with a variety of approaches, including biolistic approaches, with GIAN approaches, with electroporation, which is more classical approaches based on chemical media, magnetoffection, microinjection, which is very requires very specialized personnel and training, incubation, infiltration, which is relatively straightforward, and so on. So, in terms of the use and the potential it has in the agriculture, the biotechnology, and the food industry, phytonanotechnology serves multiple diverse purposes, including, as I mentioned before, biosensors and sensing materials. And nanosensors, in specific, enable measurement and detection of pathogens, for example. This is becoming more and more common, metabolic fluxes, as well as pesticide residues in food, so for detection purposes. And in terms of smart crop management with using tools of phytonanotechnology, it supports traditional agricultural methods by providing resource-efficient and intelligent crop management. So it doesn't negate existing technologies. If anything, it can be integrated to a more comprehensive solution. So here you can see, therefore, that the application is multi-level with benefits in both the soil level, such as improvements in soil health and fertility, in plant level, obviously, towards mainly improved growth and development, as well as stress protection against abiotic and biotic stress, but also in atmospheric content in terms of protection of environmental variables due to lower application of agrochemicals through spraying that would actually have an effect at the atmospheric level as well. So this is a preview of the review we have accepted in Trends in Plant Science. This is a figure that will appear in our review that shows the main categories of what we consider are the so-called NECs. So these are the novel engineered nanocarriers that can be used in agriculture. So of course, the main ones are based, as I mentioned, in carbon nanotubes, but you also have the CQDs, the carbon quantum dots, which are quite commonly used, so organic nanoparticles. You have graphene-based ones, like graphene oxide-based nanoparticles. You have chitosan-based ones, liposome ones, and so on. So, of course, one should not forget the importance of the inorganic ones. So you have, as I said before, the MSI NPs, the mesoporosilica NPs. You have magnetic nanoparticles, metallic nanoparticles, metallic oxide nanoparticles, and so on. Okay? And in terms of the application potential, what we identify as the main fields in the future would be effectively mainly the delivery of biostimulants. When I say agrochemicals, I also always imply in my mind the use of biostimulants, so greener technologies, of course, for protection, as well as smart delivery of nutrients. It would actually, an important field is the field of improvements in genetic breeding through advanced gene editing approaches, improved gene delivery, and of course, a never improving field is that of the biosensors, because you can easily detect, for example, with nanotechnologies, changes in ion content in salinity levels in the soil, or pH differences, or presence of pathogens, or a million different things. The possibilities are actually, I'm not going to say endless, but they're quite broad. So specifically now, focusing a bit on the categories. First of all, of course, we talked about the organic nanocarriers. So we have the chitosan-based ones, for example, and the main advantage of that is the big element of biosafety. These are bio-based, as I said before. These are biodegradable, so the load in the environment is actually preferable, with diverse applications, and they are very effective in delivering compounds like salicylic acid, melatonin, putrescine, selenium, and gibberellic acid. If you remember on my first presentation on Saturday, where I showed the research of my group, actually several of our papers are actually dealing with functionalized chitosan nanoparticles, such as mainly through the use of melatonin. If you remember, I showed you the examples of the aromatic plants under salt stress, where we also use TPP to encapsulate them in microbubbles, but we also did nanofertilization with chitosan and selenium in salt-stressed plants. So many different possibilities to actually apply this. It's okay? Okay. So the chitosan-coated fertilizers, like the chitosan conjugated with selenium I mentioned before, enhances micronutrient delivery, particularly iron, but also other elements, improving nutrient uptake. And we have an enhancement also of post-harvest treatment effectiveness, and thus extension of shelf life, which is very important, and improvement of quality of fruit. And here we have a table that was taken from a review showing major advances in chitosan nanoparticle production protocols and applications. So as you can see here that the matrices are quite variable. You can have many different matrices that are chitosan based. It's not very limited. The methodology is also, of course, quite variable, covering anything from ionic gelation to emulsion to cross-linking, which is what we usually do, coating, which we also do, as well as ionotrophic gelation. Encapsulation ingredients can, of course, also be variable. Actually, if you ask me, it could be virtually anything. And the application is indeed, as you said, also quite diverse. All of these are published works, so you can easily look them up in the literature. So in terms of the carbon-based nanomaterials, nanocarriers, you have, of course, as I mentioned before, the graphene-based ones, so like graphene oxide nanocarriers. So they're promising for the delivery of specific compounds under stress conditions. Examples include works we published in grapevine and sweet basil against salt stress specifically, but we also have functionalized graphene oxide with polyamines, if I remember well, putrescine, against heavy metals, and it was shown to be effective as well in grapevine. You also have the CQDs, the carbon quantum dots, which is again very popular, and carbon quantum dots are readily available as commercial products. It's a standard material in the Sigma catalog. They have, of course, a very small size. We're talking about less than 10 nanometers, so truly nanomaterial size, thus enabling the transport of biomolecules to distant tissues. This has also been employed in my group, having worked with it mainly in grapevines. And the functionalized CQDs boost the antioxidant apparatus, thus aiding the plant against the abiotic stressors. So our proof of function was mainly focusing on salt and heavy metal stress. And the good thing about the CQDs is that they are autofluorescing under specific wavelengths. So this autofluorescence facilitates, of course, the real-time monitoring and precise tracking, so the translocation within the tissue in a non-destructive manner, using a nice, for example, if you have a unit of confocal microscopy, you could do really nice live tracking of the translocation of the materials in the cell, in the tissue, because they autofluoresce. If they don't autofluoresce, you can actually conjugate them with specific dyes anyway, so as to be able to track them in any case. So it is still manageable. Okay? So here you see specifically the diversity that you can find in forms of carbon allotropes that can actually range, as I said before, from single-word carbon nanotubes to MWCNTs to carbon nanohorns. Many of them are just coming up on a daily basis. You think you know the literature, and somebody will come up with a new material. You have like graphite-based, diamond-based, graphitic sheet-based, nanodiamond-based, graphene dot-based, fullerene-based, and the list goes on. So as I said, imagination and good materials experts is the key to actually try to do some wonderful things. The important thing, though, I have to stress out is that you do need specialized expertise. I mean, in my group, for example, we have a very extensive presence in the field, but this wouldn't be possible without people from other disciplines. And it is something, again, that I would like to pass on as an advice, that you have to learn to collaborate with people from interdisciplinary backgrounds, because, as I say, you know, it's not possible to know everything. You know, everybody should know something really well, and that way, through collaboration, you can proceed to the best way possible. It is not a weakness not to know anything, everything. It's not a bad thing, because in Greece, for example, older professors used to think that A good paper is a paper that only has two authors, the PhD student and the supervisor. And effectively, ideally, the supervisor would only just do the proofreading at the end and just submit the paper. It doesn't work like that. Everybody needs help, you know. Anyway, it's down to the supervisor. Clay-based nanocarriers, of course, this is also very popular. And when I say clay-based, usually this covers the form of halloysite. So you can also apply functionalized halloysite clay with, for example, prolin. And we've seen in our group that it improves agronomic parameters, but also it increases secondary metabolite profiles, increased essential oil profile content in sweet basil, under salt stress conditions. So you also have the regulation of the secondary metabolic profile of the plants. And clay-based carriers show great potential in enhancing crop quality and yield through controlled compound delivery. So this is a list of the recorded advantages of using nanoclays in agriculture, which expand from the ability to encapsulate different agents that ultimately effectively leads to enhanced water absorption in the soil, to improvement of water holding capacity and nutrient release, which is extremely important, to increase in the efficiency of the input, especially the fertilizer, of course, and the water, and of course, based on the procedure you follow technologically, like the cross-linking density, the achievement of slow release pattern of nutrients that helps to control the growth over a prolonged period of time. Now, there is a great versatility to these approaches, as nanocarriers deliver essential elements to plants, and this can cover, in addition to nutrients, hormones. You can easily do that as well. As I mentioned before, the example of functionalization with putrescine or salicylic acid or melatonin, for example, osmolytes, but also amino acids such as proline, leading to improved crop health. And this actually has the potential to revolutionize agriculture by improving nutrient delivery and ultimately crop stress management. Okay, and as you can see, this can actually, the good thing about it is that there is great versatility. You can actually work at anything from the seed level to the leaf level to the root level, so foliar application or irrigation. The logic remains the same as what we discussed about in chemical priming. There's no differences whatsoever, you know. So we have the uptake of the nanoparticles, the translocation, of course, either apoplastically or symplastically in distant tissues. So it's the movement through the plant. And of course, ultimately, we're talking about the penetration into the plant cell that can actually be happening through endocytosis. It can happen through pore formation, for example. So going through stomata, if it's through foliar application, which is quite easy. It can happen through plasmodesmata, if we're talking about the symplastic transportation. So of course, this only applies for the plant cells. It doesn't apply for the animal cells. And through the function of carrier proteins. So nanotechnology does offer potential solutions to address challenges in agriculture, including adverse environmental conditions and the demand for biologically active ingredients for food. And traditional agrochemical application methods can indeed reduce the bioavailability, thus emphasizing the need we have to have a controlled delivery system of our inputs for enhanced efficacy in the plant cell. Okay? And this can improve, for example, water penetration. We can have improved increased uptake of fertilization elements and so on. So in terms of the potential applications of controlled release mechanisms using nanocarriers, this can be done by delivering biopesticides, slow release biofertilizers, by encapsulating micronutrients. I mentioned before the example of selenium and silica, for example, thus leading to stabilized plant growth. And basically also it can be used for targeted delivery of genetic material for gene editing approaches. In terms of the nano delivery systems, there is a big variety of nanodelivery systems that are developed. This includes emulsions, it includes hydrogels, as I mentioned before, like sodium alginate-based solutions, vesicular carriers, so liposomic-based, transferosomic-based, and so on. But also you can have polymeric carriers such as chitosan and chitin-based cellulose and starch and so on. And these systems offer advantages such as the high surface area, the increased activity, and the very rapid mass transfer. And these are all desirable traits for the smart delivery of the functionalized chemical. Now the other thing you should know is that a lot of them also show versatility in the sense that, for example, coatings that can be used in seeds can also be modified, the protocols, so as to also be able to deliver them in a foliar or soil irrigation context. This is done, for example, by slight chemical modifications of the protocols. In the case of sodium alginate coatings, the coating element that leads to the thin surface around the seed is achieved when adding a final step of calcium chloride in sodium alginate solution. And this leads to the solidification of the thin coating. By omitting this last step of adding the calcium chloride, you can actually maintain the sodium alginate, the functionalized form, in a liquid state, and this can also actually be applied through a liquid form, which is really wonderful because in some cases of crops, seed application is not possible. Simple as that. For those of you who remember my first presentation, I showed, for example, cases of strawberry plants where, as I mentioned, we were spraying small white unripe fruit during on-vine ripening. Okay, this solution wouldn't be possible if it was the end hydrogel solidified product. And of course, with strawberries, you cannot grow them from seed, really. There is some cases where you can obtain akins that you can grow some seeds, but it's really not done ever at a commercial level. Okay, you usually use frigo plants for that. So in that case, for example, it was imperative that you find different solutions. So indeed, you can actually modify the protocols, in this case by omitting the solidification phase, and you can therefore proceed with spraying in liquid form. So there's always ways of fixing things. You just have to all remain inventive. You have to stay proactive, keep on thinking, and solutions will always happen. And these systems, therefore, offer advantages, as I said before, that are largely desirable. And of course, in the case of post-harvest approaches, you can also do deep coatings of the fruits or vegetables. And nowadays, there is also quite trendy to carry out bi-layer or even tri-layer coatings, where you can actually play with different compositions of biopolymer coatings. I'm not really sure about the efficiency. We actually did try ourselves going for bi or even tri-layer coatings, but actually it really complicates the story. In our case anyway, we opted to go back to the single layer coating elements because the release is much more quick, actually. So in terms of the chemical strategies for the controlled release, there are different strategies that are employed for this release, including coating the materials with thin polymer films, encapsulating them in nanoporous materials, or even creating nanoemulsions. So encapsulated compounds are protected basically from evaporation and from leaching, and this ensures that we have a gradual controlled release, which is very desirable in such phytoprotective approaches. So the mechanisms for this controlled slow release, basically it is achieved mainly through mechanisms such as diffusion, degradation over time, but also response to environmental factors such as changes in pH, enzymatic activities, as well as temperature. And the release rates are indeed modulated by modifying the carrier composition, which is easy to play with. You can play with cross-linking densities, you can play with many different things. Surface properties, of course, but also the size of the nanoparticle in question. And the factors influencing nanocarrier selection, they're basically selected based on factors like, as I mentioned before, the physicochemical properties, which are variable between the different materials, the desired release kinetics. The kinetics are crucial so as to see the degree of the controlled release in your medium. Of course, initially you always do it in a in vitro setting, in a liquid medium, for example. But of course, afterwards, it is imperative that you also do that in a soil medium. So you would, for example, put your coated seeds in a soil, and then you would actually use that to obtain some sort of extracts, so as to see the amount of the priming agent that was eventually released in your setup. So it is a very tedious process, but it is an important one, so as to be able to make sure that your release will indeed be efficient. And ultimately, of course, the biology of the target plant also makes a difference. Not all species are equally receptive to the use of these materials. It is also organ specific. It is very different to have a leaf that you will spray that has a thick cuticular wax, and it's a very different thing to have one that has a very thin or even absence of a cuticular wax, such as the ones that actually grow in temperate regions that have no need for cuticular waxes, for example. And carriers can be designed for the control to release profile, of course, of the material, protection for degradation. This is really important. And this degradation could be done through UV effects, through many different factors. Improved solubility, which is, of course, extremely important, and enhanced overall cellular uptake. So there are numerous external stimuli for this controlled release. So the nanocarriers respond to external stimuli, which include factors like light, temperature, pH, as I mentioned before, but even magnetic fields for the controlled release. And external magnetic fields can be used actually to guide and localize magnetic nanocarriers, which is, though, a quite specialized approach. So in terms of the targeted delivery, in order to improve the efficiency of the end priming technology, nanomaterials offer a new delivery system mainly for what I called before improved version of next generation of chemical priming, but also for nutrition and delivery of agrochemicals. And various nanomaterials can enhance, indeed, we've seen that in the literature, nitrogen use efficiency, including nitrogen use in specific, but also phosphorus, delivering compounds effectively via the roots, the leaves, but also the seed coats, because the testa, as you know, is quite impermeable, so it is quite difficult to actually go through. So in terms of a small outline of the possibilities of the use of nanotech in towards a smart food industry, as we call it, as you can see here, this actually covers elements ranging from nano-food packaging, which deals with biodegradable or even edible packaging, for example. The good thing with chitosan coatings is that it could be actually also edible, for example, which is very important. Active and intelligent antimicrobial packaging, you can have intelligent nano-coating, use of nanosensors, and so on. You can also have implications in nano-food safety, dealing with protection of chemical deterioration, heavy metal reduction, antimicrobial activities, and so on. I'm not going to go in detail with that. The nano-food functionality, which is very important because you reach in improved quality attributes, so nutritional features of your food products, and nutritive quality. You can also have modifications in the vitamin contents and so on. And we have the enhancement, of course, of the physicochemical properties of the foods. And of course, we also have implications in nano-food preservation, and this is important because you can reduce spoilage levels, lead to humidity treatments, use of nano-preservation, and so on. So, moving away from the food industry element and going back to the phytoprotection element for the improvement of soil health in specific, nanomaterials also have the potential to improve soil health, but to manipulate rhizospheric interactions. Before, we talked about the potential of biopriming and the use of microbiomes, both on a foliar as well as soil rhizospheric level. So this is extremely important, but also potential, thus enhancing the root-soil microbiome nexus, which is a tripartite interaction that is very important towards improved crop yields. And smart delivery of these nanomaterials can alter crop responses to both abiotic as well as biotic stressors, thus enhancing the resilience to, for example, climate change-related phenomena such as drought, salinity, heat stress, and so on. So, one should also consider technicalities, technical issues that you should think about when trying to develop and optimize smart nano delivery technologies. So this also includes factors that influence the uptake and the transport of your materials in question. So mechanisms of any NEC uptake and transport in plants are generally very complex, as you would imagine. But they're influenced by the nanoparticle type, the physicochemical properties of your carrier, the plant species, and even the genotype of the species could actually be detrimental, as well as the substrate. So it makes a difference if you're talking about a soil system, a hydroponic setup, a liquid setup, or basically a culture medium. Every one of them would have different requirements. And active transport mechanisms would involve elements of signaling, as discussed previously, recycling, as well as plasma membrane, basically, regulation towards improved NEC uptake. So, in terms of the endocytic pathways in plants, which is quite similar to animals, the plant cells utilize endocytic pathways for the uptake of our novel engineered nanomaterials, and only specific nanoparticles can pass through the cell wall pores, which are quite unelastic, as you know. So basically they're semi-permeable. They are not fully permeable, but they're not fully rigid either. Okay? So in terms of the translocation, plants translocate the nanoparticles by forming complexes with membrane transporter proteins, or this could also be done through secondary metabolites that are exudated from the roots in the rhizosphere. This is also a possibility. So interaction between these nanoparticles and plants is influenced by several properties, as I said before, like porosity and actually hydrophobicity, which is also extremely important. This is why the smart choice of the material is a make or break situation. Okay? Not everything works the same. Now, in terms of the absorption of the nanoparticles by the plant, small nanoparticles, like the chitosan NP versions that I mentioned before, can be absorbed by the roots mainly through the apoplastic pathway, so never entering the living protoplast. However, larger particles are prevented from that, and absorption mechanisms include diffusion through the apoplast, crossing the plasma membrane via the symplast, and of course migration through the plasmodesmata so as to actually follow the symplastic pathway. And the large particles are actually prevented from the apoplast because of the presence of the Caspari roots. And role of xylem in the translocation is of course crucial on the upwards movement towards the aerial parts for the distribution and the translocation of these nanoparticles. So the porous network of the cell walls allows the uptake of water molecules, but also other substances that facilitates this translocation. So effectively, in simple words, the plant would actually take up the water, anything that is actually dissolved in that, and this includes your minerals, but also your functionalized nanoparticles of nanosize, and then through what we call the transpiration pathway, it would actually go upwards to the xylem so as to reach aerial tissues, as in normal water uptake and movement, which is relevant for plant nutrition. So in terms of the foliar application dynamics, contrarily, stomatal pores on the leaf surfaces serve as the main entry mechanism for nanoparticles in foliar application, as you would expect. And nanoparticles can of course penetrate the leaf tissue, while studies have indicated the translocation to other parts of the plant. And this is how and why live cell imaging is also important, through the use of either autofluorescence or conjugated dyes on these nanoparticles. And what influences these properties is basically, again, the size of the nanoparticle, its surface properties, but also the leaf characteristics. Effectively, as you know, not all leaves have the same morphology. You have such a variable morphology of leaves. You have leaves that have stomata on the adaxial surface. You have leaves that have stomata on the abaxial surface. You have leaves on the stomata that have on both surfaces. You have leaves with very specialized structures like the crypts. Crypts are basically structures where stomata are hidden within cavitations on the lower surface of the epidermis, such as the one found in laurel plants. And this is even further hidden through the presence of numerous trichomes. So there is so many different variations in leaf morphology. Even the thickness of the mesophyll would make a difference. Shade tolerant plants would have very thin mesophylls, whereas ones that actually require a lot of light would be the opposite. So leaf characteristics are crucial for this delivery and uptake. And smaller nanoparticles are actually more likely to enter through stomata. As I mentioned before, usual sizes range from 50 to 200 nanometers, when in most plant species, the stomata openings are ranging between 20 to 80 micrometers. So it is extremely easy for the nanoparticles to pass through the stomatal openings. And also, of course, you have the hydrophilic nanoparticles, the ones that are water, like the ones that like water, that can pass through the stomatal pores. Excuse me. I'll get some water. Talking for 3 hours takes its toll. So, of course, you also have the concern of the cute. We stop now? We stop now. And we will.
So we'll have lunch.
Yeah.
We'll be in an interactive session.
Perfect. We don't have a lot. Like we will finish after lunch with the closing part of this lecture, and then we'll do the IP section, and then we'll have our interaction. So we'll take care of it. So let's have a break, much needed break, and then we resume at about what time should I tell?
Resume at 2.
2. Perfect.
2 or 2:10.
Thank you.
But that last session, not the last session, is very important. IP and technology transfer, right? For any applied work that, and he has very good experience.
Yeah, I was.
aspect of priming. And then after that, I will actually do a short presentation related to IP matters, go through some terminology, some basic concepts. I have to warn you that, of course, the presentation I will make has to do with the reality I face in Europe. So I will mainly be talking about the European Patent Office and our own national agencies, as well as the US Patent Office that I'm usually exposed to. I have to admit I'm not familiar with the Indian Patent Office, so I don't know the fine details of the local IP landscape, but I will actually provide some information that would be handy if you follow a PCT application, which is a global one. Because, of course, both the EPO but also the US Patent Office are quite big offices that usually give quite global coverage. So, it's okay? Okay. So, yeah. And then, I will hopefully gain access on the internet to also open a couple of patents. So as to open a couple of patents for you to see what a typical patent includes, the files it includes, the information that is presented, and usual tools that are being employed to actually do background searches and so on. And if I understood well from Professor Sharma, tomorrow morning, although I thought today was the last exposure, tomorrow we will have a 45-minute session which will be the exchange between us in discussion of the lectures, questions, clarifications you might need, but also specific technical issues that you might have in your approaches, in your dissertations and so on. And then I think it will follow with the exam. Yeah. Okay, so good. Right. So I think we stopped there, where we were talking, of course, about the cuticle as a natural barrier. And of course, the epicuticular waxes and the cuticle do act as a primary natural barrier against the nanoparticle entrance, protecting the leaves from water loss, but also uncontrolled solute exchange. So, in order to pass through the cuticle, you have to be less than about 5 nanometers. Okay? 5 nanometers is extremely small. So, most of the conventional nanomaterials produced for agriculture practices are actually not that small. So cuticular entry is not always assured. It is possible. Like with chitosan nanoparticles, there is cases of preparations where you can go lower than that. So it could actually penetrate even through the epicuticular wax surface. However, it is not a given. It is not a fact that this will be achieved necessarily. So the role of surface modifications is that modifications or coatings on nanoparticles impact their interaction with leaf surfaces, and of course the subsequent uptake of the material. And the inclusion of various surfactants like Twin 20 is important, is critical during spraying, because it actually enhances the cuticular and the subcuticular penetration, improving efficiency. So whenever you actually do a foliar application, always make sure you actually use some sort of surfactant, because it will really amplify the efficiency of your approach. Okay? Even if you don't, it will. If you do, it will improve your observations and your data. Even if you don't, even if you come up with a solution, excuse me, for a potential industrial partner that might be interested in your idea, at the end of the day, a big part of the technology is the formulation optimization. So even if you have a wonderful active ingredient or a syncom or whatever that works nicely against some type of stress, always the end product, the commercial version of the product, will likely be quite different. There's a whole series of additions that are done so as to actually reach a final form that will most likely include such additives and surfactants for the product to reach the market. And of course, a very important part to remember is that the approach has to be highly concentration dependent, because nanomaterials, and especially nanoparticles, reach very easily the level of toxicity. You have the hormetic effect where you actually do achieve the positive effects in lower concentrations, but actually it is extremely easy to reach toxicity levels, especially with materials like metal nanoparticles and so on. Okay, so you have to be extra careful to secure that this will not be observed, the toxicity. And of course, for that, the first observation will be at the plant level, at the tissue level, using standard markers, both physiological, but actually to go more sensitive, also cellular damage indicators, to make sure that everything is behaving the way it should. Okay? So, as mentioned before, the concentration of the nanoparticles during foliar application is critical for the relocation, but the aggregation as well of the materials. So we observe dose-dependent relationships with lower doses, often exhibiting positive effects, the hormetic effect I mentioned before. While the high concentrations will most likely lead to toxicity symptoms. Okay? So with toxicity, if you have accumulation of the nanoparticles, so reaching a toxicity effect, it could be manifested in many different ways. So you could effectively have within the nucleus effects of genotoxicity and DNA damage. It is not uncommon to actually observe DNA laddering and fragmentation following high concentrations of your nanomaterials. You could even observe effects of transcriptional silencing. And then in the cytoplasm, of course, you have the blockage of ion transporters and other membrane transporters. You could have a damaged electron transport chain in the mitochondrion towards the final step of respiration. You could have alterations in photosystems 1 and 2 in the chloroplast, also affecting the light phototranspiration, basically the light photoreactions, photooxidation reactions on in the thylakoid membranes during the light reactions of photosynthesis. And you could actually also have blockage of de novo proteinosynthesis. You will have oxidation of proteins due to excess ROS being produced from the nanomaterials. And also you could get membrane damage through oxidation of the lipids of the membranes that will actually lead to ion leakage from the cell. So a lot of nasty things can happen if you actually overdo it with your nanomaterials being applied at high concentrations. And here you have a nice collective table showing information on multi-wall and single-wall carbon nanotubes and other common forms of nanoparticles like titanium dioxide, silver, iron, carbon quantum dots, and so on. Okay, and gold. So as you can see, you could have both positive and negative effects, and this is really dependent on the concentration. Okay, so you could categorize all of these toxicity symptoms in 5 different pillars, which is basically really nicely summarized here through first mechanism actually being as the dissolution, the release of the toxic ions, the second one being the size or shape dependent damage and clogging, which is highly dependent on the size. The bigger the nanoparticle, the bigger the probability of the size dependent damage. Redox cycling and Fenton reactions effects, binding interactions with other biomolecules, obviously such as proteins and nucleic acids, and as well as the oxidation and surface catalytic reaction effects in the cell. So, while applying nanomaterials in the soil, there is of course an alteration in the dynamics in the tripartite matrix that is mentioned previously between the soil, the rhizosphere, and the microorganisms. So there is a clear interaction in the root system where nanoparticles applied by soil irrigation come into contact with the root surface with the epidermis of the root and initiate therefore the uptake process from the cells. So the root interaction is critical in the uptake mechanism of nanoparticles in plants. And the major uptake route is of course through the apoplastic pathway, so effectively through the series of interconnected cell walls and extracellular spaces, never entering the living apoplast. The apoplastic pathway is therefore the main route of nanoparticle uptake in the plant roots. nanoparticles smaller than the pore size of the root epidermal cell walls, which is usually 5 to 20 nanometers, can enter through these pores. So this is also a possibility depending on the quality and the physicochemical properties of and the morphological properties, of the nanomaterials. So after root entry, once inside the root, the nanoparticles may undergo processes to reach the plant's vascular system in the central stele. So diffusion through the apoplast, movement through intracellular spaces, and the eventual arrival at the endodermis are the key steps. So here you have a very basic representation of a root dissection. And here you have, of course, the root hairs on the epidermal tissues. This is basically the epidermis, and the root is called the rhizodermis in specific. And basically the root hairs are differentiated rhizodermal cells. Okay? So as I said before, the materials, just like with water and the dissolved ions, can enter either through the symplast or the apoplast, and this is usually species dependent. In the case of the nanomaterials, as I said before, most cases this happens apoplastically, and then depending on the root, if it goes symplastically, it will move between cells through plasmodesmata, of course. If it goes apoplastically, it will actually reach a point where you will have the endodermis, and there you will find the Casparian strips. And of course, the Casparian strips are impermeable to water, so there they will have to go through the intermediate cells around the endodermis. This is basic physiology and morphology. I'm sure you're all aware of this. Thus entering the central stele, where you have the vascular tissues, so that they are loaded in the xylem vessels in order to be transported through the transpirational path. Okay? So this is another representation of the above mentioned scenarios. So in terms of the integration into the stele, where we have the vascular tissues, for nanoparticles that reach the vascular system, they must possibly integrate into the apoplast, cross the endodermis, of course, and then enter the stele. Okay? And the xylem, which is crucial, of course, in the vascular system, plays a significant role in the distribution and translocation of these nanoparticles. So xylem vessels serve as a primary pathway for uptake of water, the uptake of solutes, ions, important for mineral nutrition, but also for the nanoparticles from roots to shoots. And in terms of seed priming dynamics, specifically of nanoparticles in plants, when you have approaches where you carry out seed coatings with nanoparticles, this would involve the application of nanoparticles to the surface of the seeds, of course, and this method would allow nanoparticles to penetrate the seed, the testa of the seed, thus influencing various plant tissues during germination, but also seedling establishment. So in a diagrammatic representation of the procedure, you can see that you have the seed nanopriming with your stimulant, biostimulant, chemostimulant coating agent. A very important element of a successful seed priming technology that is based on a coating, for example, is storability. How long it can actually withstand potential storage. Okay? And this should ideally happen without dangers of premature germination. So often companies, when they look for novel technologies of seed priming, the first thing they will ask, for example, is, can we store it either at room temperature or most likely in the fridge, and for how long, without actually losing activity. Okay? So one of the main things you have to do in such approaches is to actually check the storability.
Orthodox or recalcitrant seed?
Excuse me?
Orthodox or recalcitrant seed, we have to maintain on storage.
I'm sorry, I'm having a difficulty to understand. Maybe it's because of the mask, I'm sorry.
Storage, we have to basically we have to check whether the seed is orthodox seed or recalcitrant seed for storage.
Yes. Basic analysis. I mean, it's as simple as literally just the analysis. Effectively, I'm not talking about going in depth molecularly or in terms of the seed viability. It's more in the sense of empirically or phenotypically checking whether you will get improved protection. So literally just checking whether storing your seeds in the fridge at 4 degrees for a month, 2 months, 4 months, 6 months, and so on, would actually drop. The basic thing you can do is just check germination index or something like that. Very basic stuff, you know, to begin with. And secondarily, of course, you can go downstream in a full-scale setup of the experiment to see the effectiveness of the agent, you know, in any case. And the truth is that in most cases, prolonged storage does affect the efficiency, especially if the agent in question is something like a vitamin or a hormone or something like that. You know, in this case, it does really affect it. In other cases, there is stability, but there you have to be careful with the storage. So usually you store the agents either if it's powder at room temperature, but making sure that you have silica for desiccation, I mean, so as to remove potential humidity, or if you actually store it in minus 20 degrees your material, you just have to make sure that everything is done nicely, like in standard practice, you know. And if you do that, the other thing is that many of these compounds are usually light sensitive. So the other thing is to make sure that usually you don't have a source of light that might actually break down your chemical structure. If you take care of these things, usually it's okay, because the seeds are quite, you know, they're naturally dormant in any case. So if you have the proper conditions to store them, it shouldn't be really an issue. With nosh aspirin, for example, I was really surprised to see that, okay, when we use very fresh crystals, we get super results, like really nice results. One time, we actually found accidentally a stock of crystals that was in the minus 80 minus 20, I mean, sorry, minus 20 for 7 years. We had forgotten them. And actually it was really frustrating because there was a period we wanted to do experiments and we thought we didn't have any nosh, and it turns out that we had some that was just hiding in one of the shelves. And we actually carried out an experiment and efficiency was dropped. It was not as effective as fresh material, but we would still see protective effects. And for the efficiency, for sake of time, for you not to waste time, you can also do it in vitro. What we do usually, we don't really test the efficiency of stored material, growing plants in pods and going and wasting time. It's really easy. You just get some seeds, tomato seeds or cucumber seeds or what have you. You put it in vitro plates, in petri dishes, I mean. Okay? You will add your stressor, you will add some salt or some sorbitol or mannitol or something like that, and you do a basic germination assay under stress conditions. You just check how this works. Simple as that. And actually you can get very nice, quick, easy indications of whether your viability has dropped or not. Okay? So yeah, it's important though. So you need the seed protection over time. Of course, then you have the element of the plant establishment where we see improved seed germination and plant growth. The plant tolerance to abiotic and resistance to abiotic stress is also, of course, should often be examined. And in any case, this should be done in a sustainable manner. So we have different various categories of sorry. We have both the naked form, or as I call it, of the active nanomaterials, nanoparticles, where you actually use directly your titanium dioxide nanoparticles or CQDs or what have you for direct improvements in seed germination, in activity against stressors, and so on. And the nanoparticles in this case that can be taken up and are retained by the seed coat, of course. But of course, you also have the scenario I mentioned before where you actually use them as nanocarrier systems for smart delivery and sustained release of something different, of your chemical of interest or of your microorganism or what have you. At the same time, of course, also maintaining the relative activity of the material itself. So it is also kind of an additive focus, you know. So active nanoparticles with an active compound that is loaded, which could be, as I said, nutrients, thus going to a nano fertilization, nano nutrition approach. You could use agrochemicals, like the strobilurins that we mentioned previously, essential oils, plant extracts, hormones, you name it. Actually, the stuff you could add is pretty diverse. You just have to have an imagination. So in this case, the nanoparticles are taken up and retained again by the seed coat, and then you have the slow release in the rhizosphere, depending on the cross-linking though. When I say slow release, this is also down to the nanocarrier. In the case of the nanoparticle, of course, it is direct. If you use polymers, then you can play with the cross-linking densities so as to actually make sure that you can control the rate of breakdown of the material. And in terms of the seed coat interaction, nanoparticles adhere to the seed surface or penetrate the seed tissue when used as a seed coating. And the testa, composed of multiple layers forming a physical barrier, is a potential mechanism, of course, for the nanoparticle penetration. So nanoparticles may interact with and penetrate the seed coat through microcracks, which is very common, especially in specific species, through pores, through gaps. And this could naturally appear on the spore surface of the testa, or it can be even the result of processing procedures. Okay? And factors that influence this penetration, there are several studies. I just mentioned one here as an example that have revealed that the nanoparticle concentration and the ionic strength play crucial roles in influencing the shoot fresh weight during seed priming, for example. So concentrations beyond usually 50 mg per liter showed a significant deceleration, a stoppage in nanoparticle uptake. Okay, so going really high, you have both effects, negative effects on uptake, but also possibility increased toxicity in any case. And zeta potential and the hydrodynamic diameter of the material did not notably affect uptake, thus emphasizing the importance of the biological impact of the material. So in terms of natural openings for uptake, because as I said, you can get natural gaps or cracks or pores, provide another mechanism for nanoparticle uptake. So you could have lenticels, for example. So stomata, which are normally found on leaves, of course, surprisingly, could sometimes even exist on specific seed coats. This is, of course, not really that frequent, but it does happen, providing direct access for nanoparticles to enter the seed. Okay? Actually, as time goes by, I get more and more surprised as to where I have found stomata. I mean, things that I didn't really consider in the past. I remember a few years ago, we were carrying out morphology lab practicals, and we were doing epidermal peels of onions. So we were getting like the onions, it was red onions, so as to be able to see the dye on epidermal peels under the microscope in the protoplast, and I would actually see stomata on the peels of the onion or in fruit, which is very common, of course, so because usually, people, when they think of stomata, they think of leaves, and then that's it, but that's not the case. In terms of imbibition processes, seed priming involves pre-soaking, of course, or pre-germination treatment in solutions, but of course will include your nanomaterial suspension. So nanoparticles can enter the seed during the imbibition, where water and solutes are absorbed, thus allowing for the uptake of the material. And once inside the seed, the nanomaterials interact with internal seed components, which could include proteins, could include lipids, could include cell membranes. And this interaction will, of course, alter the seed metabolism and the germination process, hopefully to the better. And here there is another diagram. Actually, again, I've taken this from a recently accepted review in Trends in Plant Science. And this is effectively an overview of the concept of the use of smart nanocarriers for improved priming approaches in agriculture. And here we actually tried to summarize everything in one figure, So the legend of that is 2 pages long. Effectively, briefly, I mean, the story, as you can see, is very complex. So of course you have the different classes of the chemical priming agents, and of course in a biased manner, as you can see, we actually mainly include here agents that we've worked in my group practically and published in the past, so mainly working with melatonin, reactive oxygen, nitrogen, and sulfur species, and noshaspirin. Here we have a summary of the different types of nanocarriers, focusing mainly on ones that we have worked with before, such as the quantum dots, the nanocrystals, and the nanopolymers. Of course, this will then, the mixture of the two will be the one that will actually lead to the functionalization of the nanocarriers for the priming agent cargo. Then, of course, the application method could be quite variable, so most likely we would go for the seed coating approach for the reasons mentioned in previous lectures. But of course, they can be done through foliar spraying or soil fertigation. But here I would like to also stress the attention that nanomaterials and nanoparticles are especially promising for biotic stress studies. Before I mentioned the element, for example, of xylem-borne diseases, right? And I mentioned verticillium. Okay? Actually, I don't know. Especially when it comes to such diseases, a big problem of that is seen in woody crops, fruit crops. So grapevine. In Europe, we have huge problems of verticillium and xylem-borne diseases in grapevine. Okay? Or trees, fruit trees. Olives, we have huge problems with Xylella fastidiosa. It is completely destroying the olive tree production. In Italy, you will be driving, which is a beautiful tree. I mean, I don't know if you've seen olive trees, but they're beautiful trees of historic importance, especially for Greece. You would be driving for hundreds and hundreds of miles in Italy, and all you would see is dead olive trees. It's completely devastating the olive production, Xylella. Actually, these approaches, I think, represent very promising ideas. So using nanoparticles, functionalized nanomaterials, to fight off these xylem-borne diseases. But one way to do that is effectively also through trunk injections. It's for direct application. You would go and you would directly inject your solution with your functionalized nanomaterials in the trunk, within the vascular tissues, for, direct delivery. And then this would be translocated and hopefully, fight off the stressor. So this is generally an approach that is not really that well researched, but I think has huge promise and potential for a specialized field of stress. But at the end of the day, we are preachers of the seed coating movement, largely, of course. Any of those is valid approaches. And there we also summarize the main methods of uptake and translocation within the leaf, as previously discussed in the lecture. Again, I will repeat that nowadays we have wonderful tools of imaging that will allow the in vivo imaging for the real-time delivery of the materials, and then this will be taken up and translocated for final activity. So, for the bioimaging that I mentioned before, either through autofluorescence of some of these molecules or by conjugating fluorescent dyes so that you actually detect them through that, electron microscopy is really advancing. As an example, I tell to my students on how things are advancing. It was only in the 1960s that we actually understood what the cytoskeleton looks like. Before the 1960s, which is not that long ago, there's professors of yours that are possibly, were around there at the time. And before that, they thought that the internal part of the cell was just completely fluid. Everything was just floating around. They didn't really comprehend the notion of a cytoskeleton that actually holds the organelles, of course, with some flexibility of movement, but holds them in a specific place. The reason why this changed in the 60s is because of microscopy. It's modern techniques in microscopy that allowed us to actually confirm that. Or another example, I will tell you about why technological advance changes what we know in books, what we read in books. In your, like for the plant biologists here, the people that know plant biology, what were you taught about whether, let's say you have a a primary growth stem. So you have a dicotyledonous crop or a monocot crop, and you don't go for the secondary growth, the primary growth of the stem. So in how do you call it? Incision, when you have like a slice of that, yes, in a section of that, would you in the center, would you actually be observing chloroplasts in the center of the stem? No. Right? This is like plant biology 101. The reason why I'm blocked now is because in the center there is a thing that I know how it's called in Greek, and I cannot think of how it's called in English. That's why I'm stuck now. In Greek we call it enderioni. It's basically the material that is in the center of the stem, of a primary growth. We call it enderioni in Greek. For some reason I got blocked now and I don't remember the word in English, how you call this material. The pith? Okay, but generally you know that you don't find theoretically chloroplasts. Okay? Think again. I actually do a whole lecture on my first year undergraduate students on morphology, where with the development of epifluorescent microscopy and the use of relevant techniques, people have discovered that especially in plants that are thriving in specific environments in the Mediterranean, like Ceratonia siliqua, acacia, and some woody tree species, for example, they discovered autofluorescence of chlorophyll in several of those plant species within the pith. So effectively saying stuff that goes completely against what you know. And there is ample microscopic evidence where you see that. Because the beauty of the epifluorescence microscopy is that it stands out. You have nice red color for the epifluorescence of chlorophyll. Okay? And this was because there were advancements in epifluorescence. And you could see that later, you know. So what we know as a norm, we found out that it has exceptions. So this is all I'm trying to say now, that probably in 20 years' time, when you have your own labs and you have your own PhD students, chances are, technologically speaking, there will be advancements that will again bring novel knowledge that will be possibly contradictory to what we know now. And this is thanks to technological advance. So in terms of the electron microscopy techniques, of course, you have scanning electron microscopy that provides a detailed view of surface morphology. You have TEM, you have transmission electron microscopy that really helps us to study the subcellular structures, for example. And of course, these are instrumental in visualizing the internalization as well as localization of nanoparticles, engineered nanoparticles, for uptake, translocation, as well as the biological impact in plants, always in high resolution. And you can use them to investigate interactions, uptake pathways, as well as I said, the cellular localization of these NENCs. So, as you can see here, it's just some examples of TEM images of nanoparticles with various compositions that you see here. You have many, many different scenarios, and as you can see, transmission electron microscopy can be a wonderful tool to actually examine the morphology of the materials, and in vitro as well as in planta, in material that it's been inoculated to. So with electron microscopy we gain significant insight, and this insight includes the visualization of, for example, silver nanoparticles and how they accumulate in plant tissues, which reveals their localization within, for example, root cells. And of course, with the microscopy, we can also determine the size, the shape, as well as the aggregation state of the functionalized nanomaterials in plant tissues. And for fluorescent microscopy, as I mentioned before, it is really useful to visualize and track NNEC movement in plants, often real time, as I said before. Fluorescent labels that are attached to NECs enable this real time monitoring and localization, although this is of course more expensive because you do need quite sensitive and expensive equipment, and even the dyes are quite expensive. So this is more of a luxury, but it's a very nice luxury for anyone who has the capacity to do that. And it provides insights into the distribution patterns as well as the cellular interactions within the cell. So advanced fluorescent microscopy advances include the use of quantum dots and other fluorophores so as to improve the sensitivity and specificity. Of course, you have the concept of confocal laser scanning microscopy, which offers improved resolution and depth penetration, simulating a three-dimensional environment in a way, which facilitates further the analysis at different tissue lengths. I don't know if you have access to confocal microscopy, but it is a wonderful tool, especially if you actually use fluorescent dyes. It is really spectacular. And as I always tell my students, of course it is also useful, but nice fluorescent microscopy is also very attractive for appearances. Sometimes, and again this is my personal opinion, I always say to my students that at the end of the day we're also salesmen. We're salesmen or saleswomen that sell a story. Our product is a story. And the story has to also look nice in addition to being of essence, you know. And this is why, through my experience, the visual component of a study could even be misleading. I mean, there's cases where you can have wonderful science that is doomed due to extremely bad presentation and writing, and you can have really mediocre science that is written in a very smart way, really souped up with some beautiful microscopy that doesn't really add that much to the story, and you know, in that way could be published in like even a decent journal. So it's all about, you know, the way you present things, and microscopy adds essence, but also adds in a qualitative manner, of course, not quantitative, but also adds what I call beauty in the manuscript. So for fluorescence microscopy, for example, it's been used to study the intracellular fate for silica-based nanocarriers in Arabidopsis that allowed to visualize their uptake and distribution within different plant cell compartments. And confocal laser scanning microscopy reveals, as I said, the internalization and the trafficking subcellularly of these NENECs in different plant species. And this has been commonly done using gold nanoparticles in Arabidopsis root cells through endocytic internalization and accumulation within endosomal compartments. So the latest advancement is, of course, the super resolution microscopy techniques, and this is covering STED microscopy and SIM microscopy. It's basically the stimulated emission depletion microscopy and the structured illumination microscopy. So these break the diffraction barrier, providing detailed insights into the subcellular localization and the interaction of the various NENCs. So they're used to study the localization and movement of quantum dot labeled nanocarriers. This is in a published example within plant cells at the nanometer scale. Okay? So stimulated emission depletion microscopy visualizes the uptake and the trafficking of polymer coated magnetic nanoparticles. This is another example that was published in literature utilizing STED in maize root cells, resolving subcellular distribution and interactions with specific organelles. And of course, we already talked about this in the past. We always have in our mind, at the back of our heads, potential environmental concerns, health and safety concerns, especially when dealing with nanoparticles. The public perception is key. I kept on talking about this, and I will talk about this again. How people perceive and accept something makes or breaks a technology. Either we like it or not. Either if it's fair or if it's not. Okay? So acceptance is key. So public concerns about unknown health effects of nanomaterials may, of course, influence consumer behaviors. And transparent communication and education on safety assessments and benefits are therefore crucial to build trust. Otherwise, you lost the game. And of course, for that, there is all sorts of legislation and regulatory frameworks, as there should be. So, of course, the nanomaterial usage falls under several different regulatory frameworks, like REACH, the CLP legislation, and GIAN. GIAN is the European Union Observatory for Nanomaterials, and it's quite a strict regulatory body that actually really monitors such research. I don't know if there is, I assume there is a relevant agency in India for monitoring the use of nanomaterials, but this is a necessity that is rightly so involved. And obligations under the REACH principles include ensuring that nanomaterials don't pose risks to the human health, but also the environment. Okay. There's also been some amendments to the REACH principles that enhanced registration requirements for the nanomaterials. And since the last 3-4 years, the registration dossier, because an interested party submits a dossier of information for the registration, requires basically much more detailed information regarding safety and compliance. So things have become more strict, in simple terms, in the last 3 or 4 years. And this is the roadmap that is actually being followed. This is taken from GIAN. This is from the public agency monitoring nanomaterial use in the European Union. So it shows the nano-relevant directives and regulations of the EU. And as you can see here, that it is a constantly dynamically evolving procedure that has been slowly addressing different industries, starting from the food additive industry, which is the more directly linked industry using nanomaterials, of course, moving to cosmetics, moving to food intended for young children, innovative foods, and then moving on to medical devices through biosensing technologies and so on. Okay? And this keeps on being further modified. So there is, of course, in closing now, in the last slide, the concept of the fate and transport in the environment. So we have to assess how nanomaterials enter the soil, the water, as well as the air. All of these components are crucial. And evaluating the potential adverse effects on non-target organisms. So basically we're talking about soil microorganisms, aquatic organisms, beneficial insects, pollinators. All of this is extremely important. And understanding nanomaterial accumulation in organisms and movement through food chains is, of course, vital to assess long-term impacts. Okay? You have, of course, the concept of the occupational exposure risks. So individuals that are involved in nanomaterial production, nanomaterial handling, but also the application should be particularly aware of potential exposure risks related to the work environment, So implementation, actually using appropriate safety measures is crucial, of course. And in terms of the health risks that can arise from nanoparticles, they're usually correlated with inhalation, also with direct skin contact. This is another obvious parameter, especially, of course, if the nanoparticles in question penetrate the biological barriers or actually are known to cause toxic effects. Okay? So assessing the potential toxicity to humans, including effects on cells, on genes, on organs, is crucial to understand the final safety profile. It is actually a requirement for the REACH dossiers that are actually submitted towards assuring registration takes place. And in terms of what you do to secure that things will go right, you actually have to build a solid risk management strategy. So it's important to implement safe by design approaches in nanomaterial synthesis. Actually is done, the synthesis, which is usually done using in silico tools, like computer programs basically, is done trying to increase potential biosafety measures through the design procedure, of course. There is, of course, application of protective measures during as well handling and use, and consideration of relevant regulations and guidelines related to the nanomaterials. So in terms of the safe by design approaches that I mentioned before, there is an integration of safety considerations during the design and development of the nanomaterials, and also a proactive strategy to minimize the risks and to enhance the overall safety of these nanomaterials. And of course, at the end of the day, you always have to be fully compliant with regulations and guidelines, especially at the regional and national level, but depending on your aspirations, also potentially more international levels. So you have to adhere to to established regulations and guidelines, thus ensuring responsible and ethical practices, of course, in the development and use of nanomaterials. So this is a very important aspect that really has to be handled. And with this, this was the end of the lecture series of the course. As I said, we will have an opportunity to spend about an hour tomorrow for feedback, questions, and anything else. I mean, we'll be very happy. Now, apparently, I must continue. Professor Sharma told me that I will talk briefly about IP matters. Is any of you, of course, this is great for you to be exposed to this, but is any of you working on any technology that is protected or is planning to be protected? Are you doing in your research, in your PhDs or whatever, research that is under IP filing or has been filed under IP? Is that no? Okay. It doesn't matter. I mean, it's okay. It's still important so that you're exposed to it. Let me just. For the record, I only started learning about IP at the very tender age of 37 years old. So it's never too late. And it was out of necessity, because I always say I'm just a poor scientist. I'm just a plant physiologist just trying to make a difference. But after a point where you develop technologies that have potential, you do realize that it is imperative that you protect your ideas. Because otherwise, there is really no guarantee that your idea will actually be protected. Unless if your approach is really open to the public approach. Something that you know from the beginning that you want to develop towards open public access strategies, which is also acceptable and respectable. Of course, in nowadays capitalistic societies, this almost never happens. But it's also a necessity, because most of these technologies usually are developed within an affiliation. And the affiliation, at the end of the day, also makes an investment. You as PhD students are working for a supervisor who is employed by a university, correct? But of course, you also utilize the resources, you utilize the bench space, you use the electricity, you use the gas, you use the water, you use the equipment. So things are not as easy and straightforward and romantic as you might think, because especially younger scientists are entitled to be romantic, but romance fades away as you get older. So yes, it's good to know some basic IP, and I will leave it at that. So let's see. And after I do this small presentation, I will require access to the internet, so that, or do you want to, it's easy? Ah, perfect, perfect. Because after I do the presentation on the theoretical part to get some concepts, I will actually enter a couple of my patents to show you the structure of a patent, the paperwork that is included on what becomes public, just to get an idea of the kind of information that is actually shared in this aspect. Thank you so much.
I have set up the proxy, so you need to disconnect that before connecting to your personal internet.
Ah, okay.
I will just do that
After we're done, you'll do that for me.
I will do that.
Yeah, when we're done with this lecture, you will do that for me.
Yes, sir.
Thank you, thank you. We finished the lecture, and now we're talking about the IP.
Actually, I got a call from the director office.
Yes.
Saying that he has got a online meeting at 4.
To go
so
now.
So you can bring him today. now itself.
Guys, do you mind if I go with Professor Sharma for a bit, and I will come back to do the to continue with the IP? So we'll do it this way. It's okay? Okay. So I'll leave it. I'll leave it here. Yes. Okay. Hopefully, I won't be too long. I'm going to meet the director. Would you give it to the person there, because he will freak out if he sees I take it with me.
Okay,
and I will avoid it later. that are there, but I assume that the overall framework, I don't expect it to be so different, because overall procedures are generally relatively standardized. Some patent offices do offer some extra functionality, like special it's a sabotage. Like the Australian office, for example, welcomes a special category of claims that is not found elsewhere in other IP patent offices, I mean. But overall, I think the framework is pretty standardized horizontally. So, in terms of the patent grants, a patent is effectively a legal monopoly that is granted to an inventor for an invention by the relevant jurisdictional authority for a limited term. Okay, so it doesn't last forever, of course. A patent grant entitles the patentee to prevent the unauthorized use of the claimed invention, and interestingly, a patent does not entitle the patentee necessarily to practice the invention. Okay, so patents are, of course, territorial. This is why I asked him, for example, in the case of the department, I was told that there are patents that are awarded, but normally in a local Indian patent office, for several reasons. It's not worldwide, although you do have options to carry out PCT filings, where these are then opened to any national stage you would like. So you could effectively pursue examination in many, many different patent offices. So in Europe, however, it's a very special situation, because inventions can be protected by two statuses. Okay, you can either go for national patents, so you can grant them by the competent national IP authority in the country of interest. So me working in an institution in Cyprus, I could actually file for a patent in the Cypriot Patent Office and stay there. You know, it's not. It could be done. The other thing is that you can also, though, however, file a patent Central European, and do that by actually doing that in the EPO, the European Patent Office. And this offers protection that you can then open to specific national examinations afterwards. So in terms of a patent, I mean, here is a slide that I will go in detail after I'm done when I use the other laptop, just to open a granted patent to see what it usually encompasses. So this is an example of a US patent about the use of specific face masks, which basically has specific claims that define the monopoly, that define why this is inventionable and unique. Okay? And actually the number of claims could be variable. It could be a patent that is based on 2 claims. It could be a patent that you have 25 claims. And interestingly, when you go in the examination stage, and if you, for example, have a PCT filing, so you open for the patent to be examined in India, in Israel, in the US, and in China. I'm just saying examples now. It could be so that the examiners of each one of those offices could allow or grant claims for different claims. So basically the Chinese officer could say that, Vasily, patents 1 to 23 are okay, but claim 24 and 25 is off because of this paper, this prior art, this whatever. Okay? Whereas the Israeli patent officer could basically say, I'm just saying, claims 1 to 20 are okay, 20 to 23 are not, and 24 and 25 that the Chinese examiner said are not okay, they are okay. So it's also down to the examiner's evaluation. And of course, this is also a procedure where you can also provide feedback or dispute. Or if an examiner says that I think this is not really patentable based on X paper, Y paper, Z paper, or X prior patent that is existing, actually you could go through the prior art and you can dispute that the claims are actually not relevant to yours. If you understand what I'm trying to say now. Yes? For example, in my case, which I will show afterwards, examples of my patents. With NOSH aspirin, for example, as I mentioned several times, the whole point of the compound, so it's a utility patent, as you will see. It's the use of something patented in a different setup, so plant use of this anti-cancer already proprietary drug. Basically, what was I going to say now? I was trying to say something and I completely forgot while I'm saying it. It's incredible. NOSH aspirin, NOSH aspirin. If I don't remember, I will continue with the hope I will remember afterwards. This shows that I'm tired. Or that I need more coffee. I don't know. Anyway. Oh, this is annoying me now. What did I want to say? NOSH aspirin, NOSH aspirin. Yes, thank God. When we did the claims in the US Patent Office, for example, it opened for examination. So initially the examiner came back with arguments, with disputes. And one of them was, if you remember, it's a simultaneous donor of NO, H2S, and aspirin. And there were claims that were based on specific references, published papers, that were using individual donors. So there was one reference of a paper published, I still remember it, in plant physiology and biochemistry, that they were using sodium nitroprusside towards protection against abiotic stresses. And the point was that this is not the same story, because using the hybrid donor that simultaneously donates the compounds would lead to synergistic effects that are more pronounced compared to the individuals. So prior art of NO priming effects in plants does not really constitute prior knowledge relevant to our own claims, if you understand what I'm trying to say now. So the claims often can be disputed. It's not that an examiner is basically a person who reads the literature. It's not like God, it's not like a person that if they say something, that's the end of the world. It's something that can be easily or not easily, you can attempt to dispute the claims if you think that there is no validity in the points. It doesn't mean that if the examiner of a patent office claims a claim is not valid, that's the end of the story. You can make a valid argument against that. This is what I'm trying to say now. Sorry? Yes, of course. And this is actually a very common practice. It's a very common thing to happen. It happens quite routinely. So in terms of the patent subject matter, there is of course the technical requirements that you have to actually show to fulfill, and you have to show an inventive step. So that should overall be deemed as patentable as an approach. Whereas if you have a mere abstraction and you're lacking sufficient inventive content in the claims, this would be rendered unpatentable. Okay? So examples of what is unpatentable subject matter is, for example, things that already exist in nature. So you cannot patent a gene. You cannot patent a microorganism. Let's say you actually have your microbial biological priming approach and you isolate some lovely bacterial strains that are only indigenous to India. It doesn't mean that you can go and you actually can patent the specific microorganisms. You could actually come up with a nice formulation with technical detail on the application, combination with surfactants or other compounds that actually further improve the activity, and this could potentially be patentable. Saying I patent this indigenous microorganisms that I discovered is not. Okay? What you could do in this case, if it's a new bacterium, you can name it for the sake of vanity, but not really patent it. Or for the genes, as I said, because people have before tried to actually go for filings for that, and this is clearly not something that is patentable. And of course, something like a mathematical formula that you come up with. So novelty is key. An invention must be new in light of the prior art. And in terms of prior art, we call prior art any evidence that an invention is already known. Okay? So prior art does not need to exist physically or, of course, to be commercially available. That's not really how it works. Okay? It is enough for something to be considered prior art that someone, somewhere, sometime, previously described, showed, or made the subject invention technology in a recorded fashion, though, that it could be shown, it could be proven. You don't have to have a competing product in the market to say this is prior art. Even more could be enough to ruin your plans. You know, something that didn't end up as being commercialized, but could be recorded as having been described somewhere, you know. And this is why, for example, the first thing that your supervisors will tell you when you have a technology that is really interesting, the first thing they will tell you is that you cannot present this anywhere. You cannot take the data of something that you want to make a claim of and go and publish it in a journal. You have to keep it closed until it becomes public. If it becomes public, then it is publishable. If it is not public, then you actually render your own claims prior art, because the examiner can actually come up with not just a paper, a poster presentation in an Indian conference that could be found online. This is prior art. This is something that was shown previously to the audience. So this constitutes the material unpatentable. So when you have something, for better or for worse, you have to keep it hidden from everyone. Don't show it anywhere, because this jeopardizes the patentability of the technology. When I say it becomes public, you will file the patent, and then the patent opens for examination. At the point of opening for examination, it is available to the public to see, in a PCT filing, for example. It could eventually not be granted. But even so, even if it's granted or not, the fact that it becomes open when you have received priority of claims from the office, that's enough for you to go and publish the paper. Okay? The important thing is to receive priority over claims by filing the hidden data. When they give you this priority, this means then that you have recorded proof that I had this idea. It was recorded on June 12th, 2022, and if somebody comes up later on with the same idea, they can check the record and see who has priority of claims of a similar nature. And if this is recorded, then you can proceed with the publication. Again, the publication, you have to be careful what you show, because let's say in the claim you file, you have different components. You do your seed priming approach and you examine X, Y, Z parameter. Okay? In the paper, let's say you have cellular damage indicator data, antioxidants, and physiological performance improvement as proof of function. You cannot actually add stuff on the paper that is not in the patent, unless if you don't mind it becoming public, if you know what I mean. You can have addenda, you can have additional information added to the portfolio under filing, but anything that you publish becomes prior art. So if it is not part of your portfolio, you lost it. You understand what I'm trying to say? Okay, good. So this is very important. Prior art is extremely important, and it is what makes or breaks a claim. So that's what it's all about, you know. So a single prior art reference cannot contain all of the elements of a proposed patent claim, and hence what I said before, that a usual claim, a usual filing can have 10, 20, 30, 35 claims. You break it down. Okay? And prior art can destroy an invention's novelty by inherency. For example, where the elements of a claim are the necessary result attendant to the prior art, whether they are appreciated or not. So, in terms of the inventive step, you also have to consider the concept of obviousness. And what is this? This is quite interesting. An invention is considered to involve an inventive step if, having regard to the state of the art, it is not obvious to a person that is skilled in this art. The could-would approach. If that person could have come up with it based on his know-how and skills and technical expertise. Even that is potentially jeopardizing a filing. The potential obviousness, that if something is obvious, even if it's not recorded, if it could have been formulated by somebody who's an expert in the field, and it doesn't encompass a truly inventive step, this is a negative. So inventive steps are evaluated by the problem and solution approach, which required analysis of the invention in terms of a technical solution to a technical problem. And for inventive steps, prior art references can be combined in a way. So an inventor's awareness of particular prior art references is irrelevant in this case. And then you have the concept of enablement, where a grant is a bargain between a government and an inventor. So an Indian grant will be a bargain between the inventor, the student, the postdoc, and the Indian government. In exchange for the resulting monopoly, the inventor will disclose the invention to the public. Okay? So in Europe, an application shall disclose the invention in a manner sufficiently clear and complete for it to be carried out by a person who is skilled in this art, following these instructions. And in the US, a patent specification shall contain a written description of the invention and of the manner and process of making and using it in such full, clear, concise, and exact terms as to enable any person skilled in the art to which it pertains, on which it is most nearly connected, to make and use the same. So to be reproducible by somebody who is a skilled expert in the field. So in terms of what you would do in the pre-filing activities, another important thing to do is always keep records. I don't know how you operate in your labs, but it is imperative for all of the students to have a lab book. I assume you all keep lab books of detailed descriptions, and the detailed descriptions should include dates and detailed protocols. Okay? So proper record keeping is key. In case of disputes, this is something that they will look back to. Okay? So inventor notebooks and invention disclosures. And you have to identify inventions that are associated with the company's or the lab's business goals or research goals. And of course, you conduct a prior art search that gives you the freedom to operate. I mean, usually, you don't really go full speed and you submit something without really checking if something else exists. It's quite silly to do that, especially because of the costs that are actually implicated. Because even the filing itself, like the paperwork to actually do this, even this costs money, not just the examination and so on and so on. So usually, it always makes sense to carry prior art searches. So it's to check the literature, the available scenery, to see if your claim is indeed patentable. Now for this, of course, most likely universities will have a technology transfer office, and it is part of the technology transfer office's duties to actually somehow be involved in this procedure, either directly through the use of one of its officers, who is usually trained in the subject, or indirectly, many tech transfer offices just commission IP experts or legal firms, IP firms, that actually do this for a living. You know, and a big part of the initial search in the in the provisional filings is actually these prior art searches. We recently submitted, about 2 years ago, we submitted a US provisional filing, and actually the charge the lawyers gave us for the prior art search alone was something like $3,000 just to check the literature. So often many labs prefer to do it themselves just to save on costs, because what it is searching prior art. So effectively searching Scopus, Google Scholar, Google Patents, Patent Scope, many different search tools to see for potential prior art. And the worst thing, to make things worse for you, is that even when it's done professionally, it is very common that claims might be disputed by examiners coming up with prior art references that were somehow not picked up by your prior art search, even if it's done by companies that are paid for this task, which is, to be honest with you, infuriating. But it happens. It really does happen. You know, so it's not really definitive. It's not really you do it and you kind of go, okay, I'm safe now. There is still dangers that during the examination something will come up that was somehow missed by the person who conducted the prior art search. So the better the work you do, the more the chances are that you will be safe indeed, that no prior art will indeed exist in your approach. Clear? It's a very frustrating procedure. generally. I mean, I've personally also been very frustrated because so far I've been involved in five filings. One of them was awarded at the global level. One of them was dropped after the provisional because we did the market search and we decided against it. And 3 right now are actually under examination. It is a very stressful, long and expensive procedure. So you have to have a strong stomach for that, you know. And do some basic reading to understand the literature. Because also for me, when I first started, I'm just a poor physiologist that knows about photosynthesis and priming agents, and I don't know. And now I have to know about prior art and eligibilities and what have you. But, you know, it's important. And you have to identify filing jurisdictions because patents are territorial. So often, especially when you go applying a filing, you also have to identify what the market is. And that's why I said if you really have a gem in your hands and you think it has great potential, that's when it's really worth going for a PCT. You know, so as to go on a more global scale. Because if you go local, in my case, if I have a technology that is only filed in the Cypriot tech transfer office, where Cyprus is a nation of 1 million people, of course, you cannot really have aspirations for a global market. You know, so it depends on the potential of your research. You know, but even then, let's say in our case of the NOSH aspirin patent that was actually awarded, we filed in the EPO, and afterwards, because of costs, the EPO would cover, what is it, 27, 29 nations that are part of the European Union. This, of course, would increase the cost because each national state would have extra charges. So to lower the costs, a thing I was asked to do by the Tech Transfer Office is to identify what I thought were key markets for the technology, which is a priming-based technology. So of course, in my mind, I thought which is countries that have a heavy agricultural economy, but also are technologically advanced and would be potentially markets for my product. And then you go, it's Italy, it's Spain, it's Germany, and so on and so on. So you highlight Holland, so you highlight 5 or 6 nations that you think would be appropriate markets, and you go for protection on those. Like in my case, for example, Cyprus was never even considered, because I don't consider Cyprus like a big market for the product, being an inventor and an invention that was carried out in Cyprus. And it's not actually, you understand my point now. So where is the market is important to identify. Now in terms of types of patent protection, of course you have the utility patents, such as the one I mentioned, using NOSH aspirin for in plants. So it is a useful process, a machine, an article of manufacture, or a composition of a matter. You have design patents that are ornamental designs embodied or applied to an article of manufacture, so it's a different category. And you also can have plant patents. In the States and Australia, you can actually patent asexually reproduced plants in specific, but it's a very specific category and a very specific regional factor. In most cases, the utility patents cover such approaches, like priming approaches, for example. And of course, patents require disclosure of the invention. They have a limited duration that is actually variable depending on the nation, the national office. It requires the government evaluation, the regional evaluation, and it protects against independent creation. Whereas in the trade secrets, the subject matter disclosure is mutually exclusive with secrecy. So you don't really have a specific term as long as secrecy is maintained. Okay? The trade secret is like the recipe for Coca-Cola. It's been unknown for 150 years, and they still have it. It's not really patented. If it was patented, it would have been open to the public after 20, 30 years, and people could reproduce. In this case, it was better for them to actually keep it as a trade secret. You know, so there is really no specific term as long as you maintain this secrecy. Somehow, this is not reverse engineered or what have you. Okay? And there is no government evaluation, of course, for trade secret creation. So the trade secret would potentially be between two parties, like a company and the inventor. And that's it. You're safe. You're done. No need, no nothing, you know. So this is another possibility that really also helps with costs if you have identified a key strategic partner that would be interested in taking advantage of the technology. Okay? But you don't have protection against independent creation if somebody comes to reverse engineer and so on and so on. So, in the terms of the filing, it is you have to determine where to file. So you have the national stage application. Of course, you have the European Patent Office, but it still requires national patent filings. Hence, the example I gave you before about choosing key markets. The more extended, it is possible, but the more the cost, you know, of course. And you also have the PCT, as I mentioned before, for the global scale, that does not result in a patent, though, itself. The PCT just allows you to actually choose your targets in a global scale, you know. In our case, after the PCT, for the nurse aspirin, we opted for and actually got granted patents in China, in Australia, in Israel. I'm thinking about like non-US and European nations that were involved, that we thought were interesting for the market, you know. And you have to determine what to file. So you can file the provisional patent, which is just US. The provisional patent is a lovely tool that I also have employed, that is done in the US Patent Office, and it doesn't really get examined. The provisional means that you get priority of claims. You actually have some data, and you want to get secure priority of claims. So you will file for a provisional in the USPTO, and then you have 12 months until this claim opens, this filing opens. So within this period, you have secured a priority of claims based on the date of filing. So you will file an envelope, a portfolio of data on January 1st of 2023, and that way you have secured priority. But this is not really open for examination. You don't know if a patent will arise or not. You just have priority of claims. And within this period, you can even have addenda. You can even add more data to enrich your claims, so that when it opens, you will make a better case to actually have the patent awarded, you know? Of course, afterwards, at one point after 12 months, you decide whether it opens for examination or not. If it opens for examination, you reach the non-provisional stage, which is an extra step, extra costs, and then it opens for examination, it becomes public. And that's when whatever you have written in the provisional filing becomes public, and this is it. I mean, this is what is being examined claim-wise. But you have secured that you have priority. So if you submit that on the 1st of January, although it doesn't open for examination, and somebody else comes and submits on the 5th of January, you have priority of claims. So it's a very nice tool, you know, because it also gives you the time to keep it secret. It's not like as you file, it will open and then that's it. You can still work on it more if you want, you know. Or this is also good because the costs are relatively smaller. So within that period, after you do the provisional, you can start making contacts with companies. And you can start to sense the interest. If they would be interested, you go to the major players. I have this non-disclosure agreement. I will share with this. And if they show interest, then it is a leverage tool. At the end of the day, it could be that everybody says, Vasily, you might think it's a great idea, but we think it's a dumb idea. So you just drop. And you never open for examination, thus saving loads of money in the process. You understand? If something doesn't have the potential to really interest the end user, what's the point of actually progressing with it, proceeding with it? And this is a nice way to safeguard this. Am I being understood? It's clear? Okay, good. You can have the full application that opens for examination, and the PCT that is a placeholder for member nations, that you can go and say this. And the typical timeline, let's say you have an EPO application in the European Patent Office filed in September 2019. The PCT would be filed a year later, in PCT 20. The US application would be filed in March 2022, so 18 months later, basically. So the issue of PPO would be about 4 years later from the initial filing. And then you would have the applications for the national stage validation based on the markets you would want to go for. The US patent would be issued by approximately 2 years later. And all of the issued patents would expire 20 years later. Okay? And in terms of the maintenance fees, of course, if you get a patent granted, that's not the end of your troubles. After all of the money you spend to the lawyers and for the filings and to whatever, then you have the wonderful, wonderful thing of maintenance fees. Yes, because if you don't pay the maintenance fees, you lose ownership of the patent. You just lose it, you know. So that's another wonderful thing you have to worry about, depending on whether it is an EPO filing or a USPTO filing. And as you can see, over a duration of 15 years, it could be something that tens of thousands of dollars just to maintain your ownership. And of course, this is really lovely when you haven't actually managed to commercialize and you just believe in the technology. with the abstract of the technology. You have the publication number, that is the identifier you use in databases to find the application. The application date, you see that journey started in 2015, and we're only now close to potentially commercializing, knock on wood. So it only took us 8 years, and not even there. It only took us 8 years to get close, potentially, you know. And here you see other information, such as the co-inventors, the applicants. You see here the research foundation, basically the tech transfer office of the City University of New York, and my university, CUT. The ES 277 blah 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 is the patent that was granted in Spain. The AU 2015 blah 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 is the Australian one, and so on and so on. The CA is the Canadian one, the CN is the China one, and so on and so on. So there is also a link in the case of the PCTs of potentially other awarded national stage patents. And the important thing for you link. You see here with all of the chemical structures of the molecules, it's the detailed description, including materials and methods, methodology, example, what and how it's done. The full set of treatments that were applied to generate the figures on data against drought stress. So in this case, for example, you see you have the controls, you have and as you can see, the claims can be broken down into multiple subclaims. I mean, in total, there are how many? In support of your findings. Okay? See it clearly. This, for example, is a QRT-PCR and then it has more detailed spray them with glyphosate, you burn everything, and your plants are just nice and green and happy. The most famous GM crop on the planet. Okay. Now, what happens? And there again, in ethics, bioethical concerns and stuff, can you patent life, an organism, blah, blah, blah. At the end of the day, it was developed by scientists in a lab. It's not a naturally occurring plant. Okay. What happens? The company, through IP, has protected its investment, and basically you buy the product and you are not allowed to obtain seed. Every year you have to get fresh seed. Okay. What happened is that there was a case many years ago in Canada, which is like a civilized state, blah, 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 but also quite liberal, you know, where there was an accusation from Monsanto of a big producer of accepted, that it was not done intentionally. It's not like the person got it and grew it on purpose. You know what the final verdict was? He was ordered to pay royalties to Monsanto, having shown that it's non-intentional. So I will stop here. You can make, chances are, I imagine, that in a court case, chances are, without being known, it's minor.
So we see you tomorrow. Plus we have the exam, so we still have to smile. You still have to be nice to me, that's right, I forgot. You can be nasty after the exams, I guess. Oh, okay.